Hello there. Welcome to Relax with Science, where the intricate poetry of the universe lulls you to sleep. Earlier today, you performed an act of quiet alchemy, and you almost certainly didn't notice it. You ate. And in the hours since, the body you inhabit, the one that has carried you through every season and every version of yourself, took what you offered and began a transformation so elegant that the word digestion feels far too clinical to describe it. As you lie here now, the day's meal is being woven into the fabric of your being. Molecules are being disassembled and reborn. Bread is becoming the spark of energy in your nerves. Protein is becoming the strength in your limbs. This entire masterpiece of molecular engineering is unfolding right now in the dark, requiring nothing from you but the simple act of existing. The digestive system is the body's great bridge between the world outside and the world within. It is a living labyrinth, approximately 9 meters long, stretching from your lips to the silent reaches of the deep interior. Every centimeter of this path is lined with specialized tissues, performing irreplaceable work. It is so sophisticated that it possesses its own second brain, the enteric nervous system, which contains more neurons than your spinal cord and can operate entirely on its own. It is a guardian, housing the vast majority of your immune cells, and a sanctuary, maintaining a hidden ecosystem of 38 trillion microorganisms. These tiny lives are so integrated into your own that they are often whispered about as a separate organ entirely, a ghost map of life within your life. Tonight, we are going to travel through this system together. Not with the frantic pace of a textbook, but slowly, the way science becomes a form of rest when it is shared in the quiet hours. We will begin at the start and drift toward the extraordinary biology of the gut microbiome. Along the way, we will pass through the stomach, the liver, and the winding architecture of the intestines. At each stop, I will share a truth about what is happening there, a fact that I hope will feel less like information and more like a quiet recognition. Your body has been doing this for you every single day of your life. Tonight, we are simply giving that devotion a name. This is Relax with Science, and tonight, we are going to travel somewhere remarkable together, slowly, gently, with nowhere else to be. Through the 9 meters of living tissue that transform what you eat into what you are. Let the room be quiet. Let the breath slow. And let the last meal you ate continue its patient journey through the biology we are about to explore. I wonder where you are tonight, what the room feels like, whether your stomach is quiet, or if you can still taste the ghost of your dinner. Whether your day was a long climb or a gentle descent. If you feel like it, and only if you feel like it, Tell me in the comments where in the world you are right now. Just a city, a country, or the time on your clock. I read every one. It genuinely matters to me that you are there. If these words bring you any ease tonight, any settling of the body toward the rest it has been working toward all day, a quiet like on the video is a small and generous thing, and it helps relax with science find its way to someone else who might need exactly this kind of company in the later hours. If you know someone who would find something like this useful, someone who lies awake when the body should be resting, consider sending this to them. No explanation needed. Just the link. And if there is ever something that could be softer, slower, or more beautiful about what we make here, I want to hear it. This channel has always belonged to the person lying in the quiet. It has always been for you. The human digestive tract is a continuous tube, approximately 9 meters in length, from mouth to anus, and despite this enormous span, the entire system is organized around a single elegant purpose: the chemical and mechanical disassembly of food into molecules small enough to cross the epithelial barrier and enter the body's internal environment. The 9-meter figure deserves to be held for a moment because it represents one of those places where biology reveals itself. Of this 9 meters, approximately 25 centimeters is esophagus, approximately 25 centimeters is stomach, approximately 6 to 7 meters is small intestine. The greatest part of the length, approximately 1.5 meters, is large intestine, and the remaining few centimeters comprise the rectum and anus. Each section is not simply a tube of uniform tissue, but a distinct organ with its own characteristic wall structure, its own epithelial specialization, its own neural and hormonal control, and its own contribution to the overall process of digestion and absorption. The wall of the digestive tract, throughout its length, follows a general organizational plan of 4 layers. From innermost to outermost, the mucosa, the submucosa, the muscularis propria, and the serosa or adventitia. But the specific characteristics of each layer at each location are so different as to make the esophageal wall and the rectal wall, while both following this plan, look almost like different organs under the microscope. The digestive system is not a tube. It is a sequence of specialized organs that happen to be connected in a line. The mouth, the entry point of the digestive system, performs the dual functions of mechanical digestion through chewing and chemical digestion through salivary amylase, while simultaneously preparing the food bolus for safe swallowing by shaping it into a cohesive mass, lubricated by the approximately 1 to 1.5 liters of saliva produced daily by the 3 pairs of major salivary glands. Chewing, mastication, is a mechanically sophisticated process that the brain coordinates through the trigeminal motor nucleus, producing the rhythmic, coordinated movements of the jaw, tongue, and cheeks that reduce food particle size, increase the surface area available for enzymatic attack, and mix the food with saliva. The force generated by the human jaw muscles, particularly the masseter and the temporalis, can reach several hundred newtons, sufficient to fracture the enamel of poorly supported teeth if applied to a hard object, and the pattern of chewing is adjusted unconsciously by the mechanoreceptors of the periodontal ligament that surrounds each tooth root, feeding back to the trigeminal motor nucleus information about the hardness, texture, and consistency of what is being processed. Saliva, produced by the parotid, submandibular, and sublingual glands, and by countless minor salivary glands scattered throughout the oral mucosa, is far more than a lubricant. It contains salivary amylase, ptyalin, that begins the chemical digestion of starch immediately upon contact, cleaving the alpha-1 for glycosidic bonds of starch chains to produce maltose and dextrins. It contains lingual lipase, which begins fat digestion. It contains lysozyme and immunoglobulin A that provide antimicrobial defense. It contains mucins that lubricate the bolus and protect the mucosa. It contains bicarbonate that neutralizes acids. And it contains growth factors, including epidermal growth factor, that support mucosal healing. The act of eating, often completed in minutes and given almost no conscious attention, is a remarkably complex biological event from its very first moment. The swallowing reflex, the pharyngeal phase of deglutition that propels the food bolus from the back of the mouth through the pharynx and into the esophagus, involves the precisely coordinated contraction and relaxation of more than 25 muscles in less than 1 second, and is one of the most elaborate reflexes in the body. Swallowing has 3 phases, of which the first, the oral phase, is voluntary, and the subsequent 2, the pharyngeal and esophageal phases, are reflexive and cannot be consciously stopped once initiated. When the tongue pushes the formed bolus backward to the oral pharynx, the swallowing reflex is triggered by mechanoreceptors in the pharyngeal mucosa, which send signals via the glossopharyngeal and vagus nerves to the swallowing center in the medulla oblongata. What happens next, in less than a second, is a precisely timed sequence. The precision of this sequence is maintained by the central pattern generator of the swallowing center, which runs the motor program without requiring conscious input. And the astonishing fact is that an average adult performs this extraordinarily complex reflex approximately 600 to 1,000 times per day, approximately once every 1 to 2 minutes during waking hours, without any awareness that anything particularly complicated is happening. The esophagus, the muscular tube connecting the pharynx to the stomach, transports food by peristalsis, the coordinated wave of circular muscle contraction that follows the bolus and propels it downward and can move food to the stomach, even against gravity, as demonstrated by the ability to swallow while standing on one's head. The esophagus is not a passive conduit through which gravity pulls food to the stomach, but an active transport organ whose smooth and striated muscle creates the wave of pressure that drives the bolus downward, regardless of posture. The esophageal wall is unusual in that its upper third is composed of striated, voluntary muscle, its lower third of smooth muscle, and its middle third of a mixture of both, a structural transition that reflects the transition from the voluntary control of the upper esophagus to the automatic peristaltic control of the lower. Peristalsis begins immediately behind the bolus as a ring of circular muscle contraction, while the muscle immediately ahead of the bolus relaxes. The descending relaxation or receptive relaxation, opening a path for the bolus to be squeezed forward. This wave of contraction progresses at approximately 3 to 4 centimeters per second, covering the approximately 25 centimeters of the esophageal length in about 8 to 10 seconds. If the primary peristaltic wave fails to clear the bolus, as sometimes happens with larger or drier pieces of food, secondary peristalsis is triggered by the distension of the esophageal wall itself, generating additional waves that continue until the bolus is cleared. At the junction with the stomach, the lower esophageal sphincter, a tonically contracted zone of smooth muscle, provides the anti-reflux barrier that prevents the acidic stomach contents from regurgitating into the esophagus, one of the most important functional valves in the digestive system. The stomach, the J-shaped muscular organ in the upper abdomen, with a capacity of approximately 1 to 1.5 liters, when moderately full, performs both mechanical digestion through its churning movements and chemical digestion through the production of hydrochloric acid and pepsin, creating the uniquely acidic environment of pH 1 to 2 that is necessary for the activation of protein-digesting enzymes. The stomach's acidity is among the most extreme in the body. A pH of 1 to 2 means a hydrogen ion concentration approximately 10 million times greater than neutral water, and this extreme acid is produced by the parietal cells of the gastric glands, specialized cells that use the enzyme carbonic anhydrase to generate hydrogen ions and bicarbonate from water and carbon dioxide, then pump the hydrogen ions across their apical membrane into the gastric lumen through the hydrogen-potassium ATPase, the most powerful proton pump in the body. The energy required for this acid secretion against an extraordinary concentration gradient is reflected in the extraordinary density of mitochondria in parietal cells, which occupy approximately 40% of the cell's volume. The acid serves multiple purposes. It activates the zymogen pepsinogen, secreted by the chief cells of the gastric glands, into the active protease pepsin. It denatures the three-dimensional structures of ingested proteins, making them more accessible to enzymatic attack. It kills most ingested microorganisms, providing a crucial antimicrobial barrier, and it provides the acid environment required for the absorption of certain nutrients, including iron and vitamin B12, indirectly through its role in liberating B12 from food proteins. The stomach's mucosa protects itself from its own acid through a remarkable defensive system, a layer of mucus secreted by the surface mucus cells, bicarbonate secreted beneath the three mechanical movements of the stomach, receptive relaxation, mixing contractions, and the antral pump, coordinate to transform the ingested food into chyme, the semi-liquid mixture of partially digested food and gastric secretions that is delivered in controlled aliquots through the pyloric sphincter into the small intestine. The stomach's mechanics are more complex than simple churning. When food enters the stomach, the fundus, the dome-shaped upper part, undergoes receptive relaxation, a vagally mediated reflex that reduces the tone of the fundic smooth muscle, allowing the stomach to accommodate the arriving food without a large increase in pressure. This relaxation is so effective that the intragastric pressure barely rises, despite the addition of several hundred milliliters of food. The food then sits in layers in the fundus and body of the stomach, with the most recently ingested food forming the outermost layer, and the oldest food moving toward the antrum. The mixing contractions, peristaltic waves that originate in the upper body and sweep toward the antrum, grind and mix the food with gastric secretions, progressively breaking down the food particles and producing the increasingly homogeneous chyme. The antral pump is the most powerful phase, strong peristaltic contractions in the thick-walled antrum squeeze the antral contents toward the pyloric sphincter, but the sphincter is almost closed, so most of the material is retropulsed back into the body of the stomach, creating a turbulent mixing that further breaks down particles. Only small aliquots, approximately 2 to 4 milliliters per contraction, are delivered through the pylorus into the duodenum with each antral contraction. The rate of gastric emptying, how quickly the stomach delivers its contents to the duodenum, is regulated by feedback signals from the duodenum. Fat, acid, and hypertonic solutions in the duodenum trigger hormonal and neural signals that slow gastric emptying, ensuring that the duodenum receives no more than it can process at any given moment. The small intestine, approximately 6 to 7 meters in length, divided into the duodenum, jejunum, and ileum, is the site of virtually all nutrient absorption, and its surface area is amplified approximately 600-fold above that of a simple tube by 3 levels of structural specialization: the circular folds of Kirkring, the villi, and the microvilli of the brush border. The challenge of absorbing all the nutrients from a meal into the body requires an absorptive surface of enormous area, concentrated in a structure compact enough to fit in the abdomen. The small intestine solves this problem through 3 nested levels of anatomical amplification. First, the plicae circulars, The circular folds of Kirkring are permanent folds of the mucosa and submucosa that project into the lumen and increase surface area approximately threefold. Second, the villi, finger-like projections of the mucosa, each approximately 0.5 millimeter to 1 millimeter tall, covering the inner surface of the entire small intestine, like a carpet of densely packed fingers, increase surface area approximately tenfold above the folded surface. Third, the microvilli, the brush border of microscopic projections that cover the luminal surface of each enterocyte, visible as a fuzzy surface coating on electron microscopy, increased the absorptive area another 30-fold. The combined amplification factor of all three levels is approximately 600, which expands what would be the roughly 5 square meters of a smooth tube into the approximately 30 to 40 square meters of the actual small intestinal absorptive surface, an area comparable to the floor of a small apartment. This surface is replaced entirely every 3 to 5 days, the enterocytes migrating from the crypts of Lieberkühn at the base of the villi up the villus surface as they mature and acquire their transport proteins, and being shed from the villus tip into the intestinal lumen as they age. The duodenum is the digestive system's most chemically active junction, the place where the three major digestive secretions converge on the arriving chyme, the gastric acid, and partially digested food from the stomach, the alkaline pancreatic juice from the pancreatic duct, and the bile from the common bile duct, both entering through the ampulla fata at the major duodenal papilla. The pancreatic juice, approximately 1 to 2 liters per day, contains bicarbonate in concentrations that can exceed 120 millimoles per liter, substantially higher than the plasma bicarbonate, making it among the most alkaline secretions in the body, and its primary function is to neutralize the gastric acid that would otherwise inactivate the pancreatic enzymes. The duct cells of the pancreas produce this bicarbonate-rich fluid through a mechanism involving the CFTR chloride channel and the sodium bicarbonate cotransporter, the same CFTR that is mutated in cystic fibrosis, which is why patients with cystic fibrosis develop progressive pancreatic insufficiency as the duct secretion becomes thick and obstructs the pancreatic ducts. The arrival of acid chyme in the duodenum triggers the release of secretin from the S cells of the duodenal mucosa, and secretin is the primary stimulus for pancreatic bicarbonate secretion, a hormonal feedback loop that precisely matches the alkalinizing response to the acid load entering the duodenum, maintaining the luminal pH in the range required for digestion. The pancreas, the dual-function gland, lying posterior to the stomach, produces approximately 1 to 2 liters of exocrine secretion per day, containing more than 20 digestive enzymes that collectively target every major class of food molecule: proteases for proteins, lipases for fats, amylase for carbohydrates, and nucleases for nucleic acids. The exocrine pancreas, the portion responsible for digestive secretion, comprising approximately 85% of the gland's mass, is organized into acini, clusters of pyramid-shaped acinar cells, whose apical surfaces face a central duct lumen, into which they secrete their enzyme-containing zymogen granules. The proteolytic enzymes, trypsinogen, chymotrypsinogen, proelastase, procarboxypeptidases, are all secreted in inactive zymogen forms to prevent the pancreas from digesting itself. The critical activation step occurs in the duodenum. The brush border enzyme enterokinase, or enteropeptidase, cleaves the activation peptide from trypsinogen to produce active trypsin, which then autocatalytically activates more trypsinogen and also activates the other zymogens in a cascade. This design, where all activation requires the duodenal enterokinase as a trigger, is the body's safety mechanism against premature activation of proteases in the pancreas itself. When this safety mechanism fails, as in pancreatitis, triggered by gallstones, alcohol, or other causes, the zymogens become activated within the pancreatic tissue, which then digests itself, producing the intense abdominal pain, inflammatory response, and potential life-threatening complications of acute pancreatitis. The pancreatic lipase, secreted as an active enzyme, not a zymogen, is the principal enzyme for fat digestion, cleaving the fatty acid chains from the first and third positions of triglycerides to produce free fatty acids and monoglycerides. Its activity requires cholipase, a cofactor also secreted by the pancreas that displaces the bile salt coating from fat droplets and anchors lipase to the droplet surface. Bile, the yellow-green secretion produced by the liver at approximately 500 milliliters to 1 liter per day, stored and concentrated in the gallbladder and released into the duodenum in response to cholecystokinin, is not a digestive enzyme, but a detergent system that emulsifies dietary fat into small droplets, dramatically increasing the surface area available for lipase digestion. Bile is one of those biological innovations that solves a fundamental physical problem. Fat and water do not mix. The pancreatic lipase is a water-soluble enzyme, and the fats it must digest are water-insoluble. Without a means of dispersing the fat into the bile salts insert their hydrophobic faces into fat globules and arrange themselves around the fat surface with their hydrophilic faces pointing outward, creating a water-compatible coating that allows the fat globule to be dispersed by the shear forces of intestinal motility into much smaller droplets, a process called emulsification. Each smaller droplet has a much higher surface area relative to its volume, providing dramatically more surface for lipase to work on. The bile salts also form micelles, small, spherical aggregates of bile salts with their hydrophobic cores pointing inward that incorporate the products of fat digestion, monoglycerides and free fatty acids, and carry them to the intestinal brush border for absorption. Without bile, fat digestion and absorption are severely impaired, producing the fatty, pale, floating stools of steatorrhea and the deficiency of the fat-soluble vitamins A, D, E, and K that depend on fat absorption for their uptake. The liver, the body's largest internal organ, weighing approximately 1.5 kilograms, performs more than 500 distinct metabolic functions simultaneously, including the synthesis of plasma proteins, the detoxification of drugs and metabolic waste products, the regulation of blood glucose through glycogen storage and gluconeogenesis, and the production of bile. The liver's functional complexity is extraordinary enough that there is no artificial device that can fully replicate it. The liver dialysis machines, available in intensive care units, can perform a fraction of the liver's functions for a limited time, but they cannot replace it. The liver receives blood from two sources: the hepatic artery, carrying oxygenated systemic blood, and the portal vein, carrying the nutrient-rich blood that drains from the entire gastrointestinal tract, the blood that has absorbed the products of digestion from the intestine. This portal blood arrives at the liver first, before reaching the rest of the circulation, and the liver has first access to everything absorbed from the gut. Glucose absorbed from a carbohydrate meal arrives at the liver in concentrations far exceeding those of the systemic blood, and the liver absorbs this excess glucose and stores it as glycogen, smoothing the postprandial glucose peak. Amino acids absorbed from a protein meal are sorted. Those needed for hepatic protein synthesis are retained. Those needed for gluconeogenesis are converted, and the remainder are released into the systemic circulation. Drugs, toxins, and metabolic waste products absorbed from the gut, including many bacteria-derived toxins that cross the gut epithelium in small amounts even in health, encounter the Kupffer cells and hepatocytes before reaching any other organ, protecting the systemic circulation from these potentially toxic substances. The liver is, in the most literal sense, the body's metabolic guardian, the organ that mediates between what the outside world provides and what the body's cells receive. The hepatocyte, the parenchymal cell of the liver, constituting approximately 80% of the liver's volume, is one of the most metabolically versatile cells in the body, possessing the biochemical machinery to perform carbohydrate metabolism, lipid synthesis and oxidation, protein synthesis, bile acid synthesis, xenobiotic detoxification, and urea production simultaneously. The hepatocyte is, in its biochemical repertoire, the most multifunctional cell in the body, a cellular Swiss Army knife, whose organelle composition reflects the breadth of its metabolic duties. The rough endoplasmic reticulum is abundant, reflecting the high rate of secretory protein synthesis. The hepatocytes produce albumin, the plasma's primary protein, which maintains oncotic pressure and serves as the carrier for fatty acids, bilirubin, drugs, and hormones. at a rate of approximately 14 grams per day, along with most of the clotting factors, complement proteins, and acute phase reactants. The smooth endoplasmic reticulum houses the cytochrome P450 enzyme system, the primary drug metabolizing enzyme system of the body, and is particularly abundant in hepatocytes, exposed to lipid-soluble toxins and drugs, and all of these functions occur simultaneously, regulated by the hormonal and nutritional environment the hepatocyte is embedded in, adjusted minute to minute in response to feeding, fasting, stress, and the hundreds of signaling molecules arriving via the portal blood and the hepatic artery. Cholecystokinin, the hormone released from the eye cells of the duodenal and jejunal mucosa in response to dietary fat and protein in the duodenum, stimulates gallbladder contraction, relaxes the sphincter of Oddi, stimulates pancreatic enzyme secretion, and slows gastric emptying, coordinating the digestive response to a fat and protein-containing meal through a single molecular signal. Cholecystokinin, CCK, was first identified as the hormone that caused the gallbladder to contract, and its name reflects this, from the Greek for bile bladder mover. But the decades since its discovery have revealed it to be the digestive system's primary coordinator of the response to fat and protein, simultaneously triggering every aspect of the secretory response that a fat and protein-containing meal requires. The gallbladder contraction that CCK produces squeezes the concentrated bile stored in the gallbladder into the common bile duct and through the sphincter of Oddi, which CCK simultaneously relaxes into the duodenum, where it emulsifies the fat arriving from the stomach. The pancreatic enzyme secretion, stimulated by CCK through receptors on the acinar cells and through vagal pathways, activated by CCK acting on vagal afferents, delivers the lipase, proteases, and amylase needed to digest the fat, protein, and carbohydrate of the meal. The slowing of gastric emptying, CCK acting on vagal pathways and directly on the pyloric sphincter, ensures that the stomach does not flood the duodenum with more fat and protein than the pancreatic enzymes and bile can process. And CCK acts on the brain, on the nucleus tractus solitarius, and the hypothalamus, via vagal afferents, and via blood-borne CCK, acting directly on CCK receptors in the brainstem to produce the satiety signal of feeling satisfied after a meal. CCK is the meal's coordinator, the molecule that turns the arrival of fat and protein in the duodenum into a whole system digestive response. The enteric nervous system, approximately 500 million neurons, distributed throughout the walls of the digestive tract, from the esophagus to the anus, organized into the myenteric plexus and the submucosal plexus, is capable of coordinating all aspects of digestive, motor, and secretory function independently of the central nervous system, earning it the name, the second brain. The enteric nervous system contains as many neurons as the spinal cord. Some estimates suggest even more, and it is the only peripheral neural structure capable of generating complex coordinated behavior independently of any input from the central nervous system. Experimental preparations in which the gut has been extrinsically denervated, all connections to the brain and spinal cord severed, continue to show coordinated peristalsis, appropriate secretory responses to luminal contents, and reflex motility patterns. The myenteric plexus, also called Auerbach's plexus, lies between the outer longitudinal and inner circular smooth muscle layers of the muscularis propria, and primarily controls gut motility, coordinating the peristaltic reflexes. The submucosal plexus, Meissner's plexus, lies in the submucosa and primarily regulates mucosal secretion and local blood flow. These two networks of neurons use the same neurotransmitters as the central nervous system: acetylcholine, serotonin, dopamine, noradrenaline, substance P, vasoactive intestinal peptide, and many others, and contain the same classes of neurons: sensory neurons that detect the mechanical and chemical state of the gut lumen, interneurons that process this information, and motor neurons that act on the smooth muscle and secretory cells. 95% of the body's serotonin is in the gut, produced by the enterochromaffin cells of the intestinal mucosa, and serotonin is the enteric nervous. The absorption of glucose from the intestinal lumen into the enterocyte is accomplished by the sodium-glucose cotransporter SGLT1, which uses the electrochemical gradient of sodium maintained by the basolateral NACAT passe to drive glucose uphill into the cell against its concentration gradient, exemplifying the principle of secondary active transport. The absorption of glucose, the primary fuel of the body, from the intestinal lumen into the blood, is one of the most intensively studied transport processes in physiology, both because of its fundamental biological importance and because of its medical relevance to conditions including diabetes and to the design of oral rehydration solutions. The challenge is a thermodynamic one. After a carbohydrate meal, the concentration of glucose in the intestinal lumen may be very high, tens of millimoles per liter, but as absorption proceeds, the luminal concentration falls, and the enterocyte must continue to absorb glucose, even when the luminal concentration is lower than the intracellular concentration. Active transport against the concentration gradient requires energy. SGLT1, the sodium-glucose cotransporter of the apical brush border membrane, solves this by coupling the uphill transport of glucose to the downhill transport of sodium. For every molecule of glucose moved into the cell, 2 sodium ions move simultaneously, driven by the large inward sodium electrochemical gradient maintained by the NACAT passe on the basolateral membrane. The energy stored in the sodium gradient, generated by the ATP-consuming NACAT passe, is thus the ultimate energy source for glucose absorption. This is secondary active transport. The energy of ATP does not directly move the glucose, but it maintains the sodium gradient that indirectly drives it. The clinical elegance of this mechanism is exploited in oral rehydration therapy. Adding glucose to a fluid given to a person with secretory diarrhea stimulates SGLT1 activity in the enterocytes, which co-absorbs sodium along with the glucose, and the sodium absorption drives osmotic water absorption, restoring fluid balance. A teaspoon of sugar and a pinch of salt in a liter of water, used appropriately, can reverse the fatal dehydration of cholera. Fat absorption in the small intestine involves a unique pathway. The products of fat digestion, monoglycerides and fatty acids, are taken up by enterocytes, reassembled into triglycerides in the smooth endoplasmic reticulum, packaged with phospholipids and protein into chylomicrons, and secreted into the lacteals of the intestinal villi, rather than directly into the portal blood. The distinction between the route of fat absorption and the route of carbohydrate and amino acid absorption is one of the digestive system's most elegant structural solutions to a biochemical problem. Carbohydrates and amino acids, being water-soluble, can be absorbed directly into the portal blood via the capillaries of the intestinal villi and carried to the liver in the portal circulation. Fat and fat-soluble vitamins, being lipophilic, would not survive as free molecules in the aqueous blood plasma. They must be packaged into lipoprotein particles before transport. The chylomicron, the largest lipoprotein particle in the body, assembled in the enterocyte, is too large to enter the blood capillaries through their endothelial junctions, but it can enter the lymphatic lacteals, whose larger pores and button junctions accommodate it. From the lacteals, the chylomicrons travel through the mesenteric lymphatics to the thoracic duct and enter the venous blood at the left subclavian vein, bypassing the liver entirely in their first pass through the circulation. This bypass has clinical significance. Drugs and nutrients that are formulated in lipid-based delivery systems can also travel this lymphatic route, avoiding first-pass hepatic metabolism that would otherwise degrade them. Long-chain fatty acids take this lymphatic route via chylomicrons. Medium-chain fatty acids, found in coconut oil and some other foods, are more water-soluble and are absorbed directly into the portal blood without requiring chylomicron packaging, which is why medium-chain triglyceride formulas are used in conditions of impaired chylomicron assembly or lymphatic transport. The intrinsic factor produced by the parietal cells of the gastric fundus is essential for the absorption of vitamin B12. Vitamin B12, cobalamin, follows the most elaborate absorption pathway of any micronutrient, requiring 3 successive binding proteins before it can cross the intestinal epithelium. In food, vitamin B12 is bound to proteins in the animal products that contain it. In the stomach, the parietal cells secrete hydrochloric acid and pepsin that denature these food proteins, releasing the B12, which then binds to haptocorin, our binder, secreted by salivary glands and gastric mucosa. When the complex reaches the duodenum, the alkaline pH and pancreatic proteases degrade the haptocorin, releasing the B12 again. The free B12 then binds to intrinsic factor, a glycoprotein secreted by the parietal cells, and the intrinsic factor B12 complex is resistant to further digestion. This complex travels intact through the entire length of the small intestine until it reaches the terminal ileum, the last 30 centimeters of the small intestine, where the only receptors for intrinsic factor in the body, the cubolin-megalin complex, bind and internalize it. The B12 is then released inside the enterocyte and secreted into the portal blood, bound to transcobalamin. The extraordinary length of this journey through the entire small intestine means that any condition that eliminates the terminal ileum, surgery for Crohn's disease, cancer, or trauma, destroys the body's only site of B12 absorption, producing the megaloblastic anemia and neurological damage of B12 deficiency, unless supplementation is given by injection, bypassing the gut entirely. The brush border of the intestinal microvilli is not merely a physical amplification of the absorptive surface, but contains a dense array of digestive enzymes. The brush border enzymes including lactase, sucrase isomaltase, maltase, and the peptidases that complete the terminal digestion of disaccharides and oligopeptides at the very surface of the absorptive cell. The brush border enzymes represent the digestive system's final processing step, the completion of the digestion of carbohydrates and proteins at the point of absorption, ensuring that monosaccharides and amino acids, rather than their larger precursors, are what crosses the epithelial barrier. Lactase, the enzyme that cleaves lactose into glucose and galactose, is perhaps the most clinically discussed of these enzymes because its expression is regulated developmentally in most mammals, present in high amounts in infancy, when lactose from breast milk is the primary carbohydrate source, and progressively reduced through childhood and adulthood in the majority of the world's human population. Lactase persistence, the continued expression of lactase through adulthood, is actually the derived minority condition driven by genetic mutations in the lactase gene regulatory region that arose independently in several human populations that practiced cattle herding and dairy farming, providing a selective advantage for individuals who could digest dairy products in adulthood. The majority of adults worldwide are lactase non-persistent and experience the bloating, flatulence, and diarrhea of lactose intolerance when consuming large amounts of lactose, not because they are unusual, but because they retain the ancestral mammalian pattern. The sucrase isomaltase enzyme, which cleaves sucrose and certain products of starch digestion, is congenitally absent in sucrase isomaltase deficiency, a rare condition that produces carbohydrate malabsorption from early childhood. These brush border enzymes are not secreted into the lumen, but are membrane-anchored proteins whose active sites face the lumen while they remain embedded in the brush border membrane, ensuring that digestion and absorption are coupled at the same location. The ileocecal valve, the mucosal fold at the junction of the terminal ileum and the cecum, regulates the transit of intestinal contents from the small to the large intestine, preventing the reflux of cecal bacteria into the nutrient-rich ileum and controlling the delivery rate to the colon, functions whose impairment produces the bacterial overgrowth syndromes of the small intestine. The ileocecal valve is one of the digestive system's most important anatomic. The valve itself is not a simple mechanical structure, but a mucosal fold reinforced by smooth muscle that maintains a tonic closure, opening when the ileum contracts and closing during cecal distension. The functional importance of the valve lies particularly in preventing retrograde bacterial contamination. The colon contains approximately 10 to the power of 11 bacteria per milliliter, the densest microbial community in any environment on Earth, while the terminal ileum has only approximately 10 to the power of 7 bacteria per milliliter. The ileocecal valve's competence maintains this difference of 4 orders of magnitude in bacterial density between the small and large intestine. When the valve becomes incompetent, or when the normal small intestinal motility that sweeps bacteria aborally is disrupted by stasis, by obstruction, or by anatomical variance, bacteria from the colon can migrate into the small intestine, producing small intestinal bacterial overgrowth, the fermentation of nutrients by colonic bacteria in the small intestine that produces bloating, diarrhea, and malabsorption of fat and B12. The large intestine, approximately 1.5 meters in length, divided into the cecum, ascending, transverse, descending, and sigmoid colon, and the rectum, absorbs approximately 1 to 1.5 liters of water per day from the approximately 8 liters of fluid that enters it from the small intestine, concentrating the intestinal contents from a liquid to a semi-solid stool. The large intestine's primary function, water absorption and stool formation, is less dramatic than the small intestine's enzymatic digestion and nutrient absorption, but is physiologically critical. Without adequate colonic water absorption, the body would lose volumes of fluid in the stool that would rapidly produce life-threatening dehydration. Of the approximately 9 liters of fluid that enter the duodenum each day, from food, from swallowed saliva, from gastric secretions, from pancreatic juice, from bile, and from intestinal secretions, approximately 8 liters are absorbed in the small intestine, leaving approximately 1 liter to enter the colon. The colon absorbs most of this, passing approximately 100 to 200 milliliters per day in the stool. The absorptive capacity of the colon is substantial. It can absorb up to 4 liters per day when maximally stimulated. But this capacity is not usually called upon because the small intestine has already done most of the absorption. Water absorption in the colon is coupled to sodium absorption. The colonocytes actively absorb sodium through epithelial sodium channels, creating an osmotic gradient that drives water absorption and is regulated by aldosterone, the same mineral corticoid that regulates renal sodium reabsorption. The colonic mucosa lacks the villi of the small intestine. It is a flat surface, punctuated by the crypts of Lieberkühn, and its epithelium is specialized for absorption and mucus secretion, rather than for enzyme-mediated digestion. The thick layer of mucus, produced by the abundant goblet cells of the colon, lubricates the passage of the increasingly solid stool and protects the epithelium from the mechanical abrasion of the fecal mass. The gut microbiome, the community of approximately 38 trillion microorganisms residing primarily in the colon, comprising bacteria, archaea, viruses, fungi, and protozoa, is now considered by many researchers to function as a virtual organ, contributing to digestion, immunity, metabolism, and even brain function through its production of short-chain fatty acids, neurotransmitter precursors, and immune modulators. The number 38 trillion deserves to be held for a moment. The human body contains approximately 30 trillion human cells. The gut microbiome contains approximately 38 trillion microbial cells, slightly more non-human cells than human cells. The vast majority of these are bacteria, concentrated in the colon in densities of approximately 10 to the power of 11 to 10 to the power of 12 per milliliter of colonic contents, the highest density of living organisms in any known environment, surpassing the density of cells in any human tissue. The microbiome is dominated by two phyla, the firmicutes and the bacteroidetes, along with actinobacteria, proteobacteria, and varicomicrobia in smaller proportions. And within these phyla are hundreds of species, the specific composition of which varies dramatically between individuals, between populations. The bacteria ferment the dietary fiber and resistant starch that human digestive enzymes cannot break down, producing the short-chain fatty acids, butyrate, propionate, and acetate, that are the primary fuel for the colonocytes, maintain the gut barrier integrity, modulate immune function, and regulate glucose and lipid metabolism throughout the body. They synthesize vitamin K and certain B vitamins. They exclude pathogens through competition for nutrients in space. They train the immune system. And through the gut-brain axis, they influence mood, anxiety, and stress responses in ways that are only beginning to be understood. Butyrate, the short-chain fatty acid produced by the microbial fermentation of dietary fiber in the colon, is the primary fuel for colonocytes, providing approximately 60 to 70% of their energy requirements, and its production is so essential to colonic health that reduced butyrate production from low-fiber diets or dysbiosis is associated with colorectal cancer, inflammatory bowel disease, and metabolic syndrome. Butyrate is the link between dietary fiber and colonic health, the molecule through which the indigestible polysaccharides of fruits, vegetables, legumes, and whole grains are converted by the bacteria of the colon into a fuel that feeds and maintains the very cells that house those bacteria. The symbiosis is elegant. The host's digestive enzymes cannot break down dietary fiber, so the fiber arrives in the colon intact, where the resident bacteria ferment it to produce butyrate, propionate, and acetate. The colonocytes consume the butyrate avidly. It enters the cell as a sodium-coupled transport, is rapidly oxidized in the mitochondria, and powers all of the colonocytes' energy-requiring functions, including tight junction maintenance, mucus secretion, and immune modulation. Butyrate also acts as a histone deacetylase inhibitor, suppressing the expression of pro-inflammatory and pro-proliferative genes in the colonocytes, which is one mechanism by which it may protect against colorectal cancer. In conditions where butyrate production is reduced, when the diet is low in fiber, when the butyrate-producing bacteria are depleted by antibiotics or by dysbiosis, or when motility disorders prevent adequate fermentation time, the colonocytes are functionally impoverished. The tight junctions between colonocytes weaken. The mucus layer thins. The inflammatory tone rises. The integrity of the epithelial barrier is compromised, allowing bacterial products to permeate into the submucosa and the systemic circulation in a process sometimes called leaky gut, a state associated with chronic systemic inflammation, metabolic disease, and autoimmune conditions. The gastric emptying rate, the speed at which the stomach delivers chyme to the duodenum, is regulated by a sophisticated feedback system involving hormonal signals from the duodenal mucosa, the vagus nerve, and the intrinsic properties of the pyloric sphincter, ensuring that the duodenum is never overwhelmed with more acid, fat, or hypertonic solution than it can neutralize and process. Gastric emptying is not simply a matter of the stomach contracting. It is a precisely regulated delivery system that matches the rate of gastric output to the capacity of the duodenum to receive and process it. The regulatory signals form an integrative feedback loop. Fat in the duodenum stimulates the eye cells to release cholecystokinin, which acts on the pyloric sphincter and the antrum to slow emptying. Acid in the duodenum, when the pH falls below about 4.5, stimulates the S cells to release secretin, which inhibits gastric acid secretion and slows motility. Hypertonic solutions in the duodenum trigger neural reflexes through the submucosal plexus in the vagus, the enterogastric reflex, that slow gastric emptying. Distension of the duodenum activates mechanoreceptors that similarly inhibit gastric motor activity. The net result is that the rate of gastric emptying is continuously adjusted to match the delivery of stomach contents to the duodenum's processing capacity. When the duodenum is coping well, emptying proceeds. When it is overwhelmed, emptying slows. This regulation explains why the composition of a meal determines how long it takes to leave the stomach. Pure carbohydrate solutions the gallbladder, the pear-shaped sac on the underside of the liver, concentrates bile up to tenfold by absorbing water and electrolytes while storing it between meals, and the cholecystokinin-stimulated contraction that empties it during a fatty meal delivers this concentrated bile into the duodenum in a bolus that provides the immediate emulsification capacity needed for efficient fat digestion. The gallbladder is often described as the liver's storage annex for bile, but this description understates its active contribution to the bile's function. The liver produces bile continuously at approximately 500 to 1,000 milliliters per day, but the duodenum needs bile only during and after meals. Between meals, the sphincter of Oddi is closed, and bile backs up into the gallbladder through the cystic duct. In the gallbladder, the epithelial cells actively absorb water, sodium, chloride, and bicarbonate from the bile without changing the composition of the bile salts and other organic molecules, concentrating the bile up to tenfold, so that a bile that was approximately 50 millimoles per liter of bile salts in the hepatic duct may be 500 millimoles per liter in the gallbladder. This concentration is critical for the bile's emulsifying function. The concentrated bile salts, delivered as a bolus when the gallbladder contracts, in response to cholecystokinin, provide an immediate and substantial bile salt concentration in the duodenum that can effectively emulsify the fat droplets of the arriving chyme. The formation of gallstones, cholelithiasis, occurs when the balance of the bile's constituents is disrupted when cholesterol is supersaturated relative to the bile salts and phospholipids that normally keep it in solution. Cholesterol crystals nucleate and grow into stones. 80% of gallstones are cholesterol stones. Their development is promoted by obesity, rapid weight loss, estrogen, which increases biliary cholesterol secretion, and prolonged fasting, which reduces the cycling of bile salts through the enterohepatic circulation and reduces their concentration in bile. The enterohepatic circulation of bile salts, the cycle by which bile salts are secreted into the bile, pass through the intestine, are actively reabsorbed in the terminal ileum, return to the liver via the portal blood, and are resecreted in the next bile secretion, allows the body to maintain an adequate bile salt pool, with only modest daily synthesis, recycling each bile salt molecule approximately 6 to 10 times per day. The enterohepatic circulation is one of the body's most elegant resource conservation systems, a recycling loop that allows the approximately 3 to 5 grams of bile salts in the total bile salt pool to support the emulsification of the 8 to 12 grams of dietary fat consumed in an average day. Without recycling, the liver would need to synthesize enough bile salts to match the daily fat intake, a synthesis burden that would be difficult to meet, given that bile salt synthesis from cholesterol is a multi-step process limited by enzyme capacity. With recycling, the liver only needs to synthesize the small fraction, approximately 500 milligrams per day, lost in the feces, while the rest is reclaimed. The active reabsorption of bile salts in the terminal ileum is accomplished by the apical sodium-dependent bile acid transporter, ASBT, which transports bile salts against their concentration gradient from the ileal lumen into the enterocyte, from which they exit through the basolateral, organic solute transporter, and enter the portal blood. In the portal blood, the bile salts are bound to albumin and travel to the liver, where the hepatocytes extract them with approximately 95% efficiency on the first pass. The hepatocytes then reconjugate any bile salts that were deconjugated by bacterial enzymes in the colon and resecrete them into the bile. The cycle completes approximately 6 to 10 times per day, with the small fraction that escapes ileal reabsorption and reaches the colon being excreted in the stool as the daily fecal bile salt loss. The peristaltic, its fundamental organization, contraction behind, relaxation ahead, is preserved from the esophagus to the rectum, and it represents one of the most thoroughly studied neural reflexes in gastroenterology. The reflex is initiated by the mechanical distension of the intestinal wall caused by the luminal contents, which activates the intrinsic primary afferent neurons of the submucosal plexus. These neurons respond to the stretch by releasing serotonin and substance P onto the interneurons of the myenteric plexus. The interneurons then coordinate two opposing pathways, the ascending excitatory pathway, which activates excitatory motor neurons that release acetylcholine and substance P onto the circular muscle behind the bolus, causing it to contract, and the descending inhibitory pathway, which activates inhibitory motor neurons that release nitric oxide and vasoactive intestinal peptide onto the circular muscle ahead of the bolus, causing it to relax. The segment of intestine containing the bolus is thus narrowed by the contracting muscle behind and opened by the relaxing muscle ahead, and the pressure differential propels the bolus forward. The importance of nitric oxide as the primary inhibitory neurotransmitter of the descending pathway was revealed by the disorder achalasia, where the inhibitory neurons of the lower esophageal sphincter are lost, leaving the sphincter unable to relax during swallowing and producing the functional obstruction of the esophagus that prevents swallowing. The mucus layer of the intestine is a two-layer structure, an inner, firmly adherent layer that is essentially sterile and impenetrable to bacteria, and an outer, loosely adherent layer that is colonized by bacteria, and its maintenance is critical for the separation of the intestinal microbiome from the epithelial surface, with its disruption being a key feature of inflammatory bowel disease. The intestinal mucus layer is the physical boundary between the microbiome and the body, the biological fence that maintains the extraordinary proximity of 38 trillion bacteria to the epithelial surface without producing the chronic inflammatory response that direct epithelial contact with bacteria would trigger. The mucus is produced by the goblet cells of the intestinal and colonic epithelium, which constitute approximately 10% of the epithelial cells, and produce the large, heavily glycosylated mucin glycoproteins, primarily MUC2 in the colon, that form the structural scaffold of the mucus layer. In the colon, the inner mucus layer is approximately 50 micrometers thick and so tightly cross-linked that bacteria essentially cannot penetrate it. The outer layer is thicker, approximately 100 micrometers, and its looser structure allows bacterial colonization that provides the bacteria access to the mucin sugars as a nutrient source, while keeping them at a safe distance from the epithelium. The maintenance of the inner mucus layer requires continuous goblet cell secretion, driven by goblet cell autophagy, by signals from the enteric nervous system, and by microbial metabolites including butyrate, and its degradation or thinning is one of the earliest features of the colitis that characterizes inflammatory bowel disease. In mouse models of colitis, the mucus layer is thinner or absent before any other evidence of inflammation appears, consistent with mucus disruption being a cause, rather than a consequence, of intestinal inflammation. The inner mucus layer is the last line of physical defense between the microbiome and the body. Secretory immunoglobulin A, the predominant antibody in intestinal secretions, produced by plasma cells in the lamina propria and transported across the epithelium by the polymeric immunoglobulin receptor, is the immune system's primary antibody interface with the gut microbiome, coating approximately 80 to 90% of commensal bacteria without triggering inflammation. Secretory IgA is the gut immune system's most diplomatic instrument, an antibody that binds to microorganisms and antigens in the intestinal lumen without activating complement, without triggering inflammatory responses, and without causing cell death. This non-inflammatory character distinguishes secretory IgA from IgG and IgM, which activate complement when they bind their targets. The non-inflammatory quality is essential in the gut, where the immune system must prevent invasive pathogens. After production, the dimeric IgA binds to the polymeric immunoglobulin receptor on the basolateral surface of the epithelial cells, is transcytosed through the epithelial cell, and the receptor is cleaved to release the secretory IgA, now attached to the secretory component, a fragment of the polymeric immunoglobulin receptor that protects the antibody from proteolytic degradation in the harsh intestinal environment into the lumen. In the lumen, secretory IgA coats bacteria, blocking their adherence to the epithelial surface in a process called immune exclusion. It neutralizes viruses and toxins. It selectively binds to the members of the microbiome that have broken through the mucus barrier. In doing so, It shapes the composition of the microbiome. The bacteria most heavily coated by IgA are often those most likely to induce inflammation if they were to translocate across the epithelium without killing them or triggering the inflammatory cascades that would damage the gut tissue. The haustral contractions of the colon, the segmental, non-propulsive contractions that mix the colonic contents and expose them to the absorptive epithelium, are distinct from the mass movements that periodically propel fecal material toward the rectum, and the coordination of these two patterns of motility by the enteric nervous system and autonomic control determines the transit time of fecal material through the colon. The colon is not simply a passive tube that slowly moves its contents toward the rectum. It is a modal organ with two functionally distinct movement patterns that serve different physiological purposes. The haustral contractions, named for the haustra, the saculations visible in the colon on imaging, are segmental contractions that appear and disappear at different points along the colon, mixing the contents without propelling them significantly in either direction. These contractions slow the transit, increasing the contact time between the luminal contents and the absorptive epithelium, maximizing water absorption and allowing the microbial fermentation of dietary fiber adequate time to proceed. The colonic contents may remain in the ascending and transverse colon for 12 to 24 hours, during which most of the water absorption and fermentation occurs. Superimposed on these mixing movements are the mass movements, also called high-amplitude propagating contractions, that occur 3 to 4 times per day, typically in the morning and after meals. These are powerful, rapid contractions that travel from the transverse or descending colon to the sigmoid, propelling the fecal material several tens of centimeters toward the rectum in a single event. The postprandial mass movements, particularly those triggered by the gastrocolic reflex, the colonic motor response to gastric distension at the start of a meal, are why many people feel the urge to defecate shortly after eating breakfast. The gastrocolic reflex is mediated partly by cholecystokinin and partly by neural reflexes through the enteric and autonomic nervous systems, and its strength varies considerably between individuals. The liver's detoxification of drugs and xenobiotics, primarily through the cytochrome P450 enzymes of the smooth endoplasmic reticulum, follows 2 phases: phase I reactions that introduce or expose reactive functional groups through oxidation, reduction, or hydrolysis, and phase II conjugation reactions that attach large polar groups to these reactive sites, making the metabolite water-soluble and suitable for renal or biliary excretion. The liver's handling of drugs and foreign chemicals is the biochemical basis of pharmacology, the understanding of drug metabolism that determines how long a drug remains active in the body, what its active and toxic metabolites are, and what happens when 2 drugs compete for the same metabolic pathway. The cytochrome P450 enzymes, a superfamily of heme-containing monooxygenases, named for their absorption at 450 nanometers, when combined with carbon monoxide, are the workhorses of phase I metabolism. The most important drug metabolizing P450 is CYP3A4, which handles approximately 50% of clinically used drugs, CYP2D6, which handles about 25%, and CYP2C9, CYP2C19, and CYP1A2, which handle most of the rest. Phase II reactions: conjugation with glucuronic acid, glucuronidation, via UDP glucuronosyltransferases, sulfate, sulfation, glutathione, glycine, or acetyl groups, attach bulky, polar groups to the phase I product, converting it from a lipophilic molecule that would simply be reabsorbed in the kidney into a water-soluble molecule that can be filtered in the glomerulus and excreted in the urine, or that can be secreted into bile. The clinical importance of this system is enormous. Genetic polymorphisms in cytochrome P450 genes create the poor metabolizers and ultra-rapid metabolizers of certain drugs that explain why the same dose of a drug is therapeutic in one patient and toxic or ineffective in another. You have been with me for a while now, traveling through the mouth and the stomach, in the small intestine and the liver, in the microbiome, and all the quiet, tireless biology of digestion. I find that genuinely wonderful. If these nights together mean something to you, if there is something in the pace of this, in the way the science of the body becomes a kind of rest for the mind, then subscribing to Relax with Science is a simple and warm way to say, yes, I would like more of this. I would like to come back. And I will be here whenever you do. The migrating motor complex, the pattern of cyclic motor activity that sweeps through the stomach and small intestine approximately every 90 minutes during fasting, is often called the intestinal housekeeper, clearing the fasting small intestine of bacteria, debris, and undigested material that would otherwise accumulate and cause bacterial overgrowth. The migrating motor complex, discovered by Joseph Code and James Marlett in the 1970s, is one of the most important and most underappreciated motility patterns of the digestive system. During the fasting state, approximately 2 hours after a meal, when the fed state motor pattern has subsided, the stomach and small intestine enter a cyclical pattern of motor activity that repeats approximately every 90 minutes. Each cycle has 3 phases. Phase 1 is a period of motor quiescence, lasting approximately 45 minutes. Phase 2 is a period of irregular, intermittent contractions of increasing intensity, lasting approximately 30 minutes. Phase 3, the activity front, is a brief but intense burst of regular, high-amplitude contractions at the maximum frequency of the small intestinal smooth muscle, 11 to 12 per minute in the duodenum, 8 to 9 per minute in the ileum, that sweeps from the stomach to the terminal ileum over approximately 90 minutes, propelling any remaining luminal contents aborally toward the colon. This phase 3 activity front is the intestinal housekeeper. It clears the small intestinal lumen of any debris, excess mucus, exfoliated cells, and bacteria that have accumulated since the last cycle. The migrating motor complex is driven by the hormone motilin, released cyclically by endocrine cells in the proximal small intestine, and is disrupted by eating. The arrival of food in the stomach replaces the fasting motor pattern with the fed motor pattern and suppresses motilin release. The clinical importance of the migrating motor complex becomes apparent when it fails. The neuropathies, medications, opioids are particularly disruptive, and structural conditions that suppress it allow bacteria to accumulate in the small intestine, producing the bacterial overgrowth, bloating, and malabsorption of the associated syndrome. The absorption of iron in the small intestine is a tightly regulated process that distinguishes between heme iron from meat and fish, absorbed by a dedicated heme transporter, and non-heme iron from plants, whose absorption depends on its reduction from ferric to ferrous form by duodenal cytochrome B, and transport by the divalent metal transporter, and which is dramatically affected by the composition of the meal. Iron absorption is one of the digestive system's most carefully regulated processes, reflecting the particular challenges of iron homeostasis. The body has no significant mechanism for excreting excess iron, so absorption must be precisely calibrated to need. The daily absorption requirement is only approximately 1 to 2 milligrams, matching the small obligatory daily losses from shedding of intestinal and skin cells, but the diet may contain 10 to 15 milligrams per day, meaning that only 10 to 15 heme iron is generally absorbed at 20 to 30% efficiency, relatively insensitive to dietary composition. Non-heme iron from plants, fortified foods, and supplements is present in the ferric form that must be reduced to ferrous iron by the duodenal cytochrome B reductase on the brush border before it can be transported into the enterocyte by the divalent metal transporter DMT1. This reduction is facilitated by vitamin C, ascorbic acid, in the meal, which is why vitamin C dramatically increases non-heme iron absorption. Conversely, phytate from whole grains and legumes, polyphenol from tea and coffee, and calcium all inhibit non-heme iron absorption by chelating or competing with the ferrous iron. The absorption efficiency of non-heme iron from plant sources varies from less than 1% to over 20%, depending on these dietary factors. The defecation reflex, the coordinated contraction of the rectum, and relaxation of the internal anal sphincter, triggered by rectal distension, is a spinal reflex that can be consciously modulated by voluntary control of the external anal sphincter, but this voluntary control is dependent on intact sacral spinal cord function and is lost in spinal cord injuries below the sacral level. Defecation is the digestive system's most intimate negotiation between reflex and volition. When fecal material distends the rectum above the threshold of approximately 100 milliliters, the distension activates mechanoreceptors in the rectal wall that trigger the rectoanal inhibitory reflex, the involuntary relaxation of the internal anal sphincter, which is composed of smooth muscle and is under autonomic control. Simultaneously, the puborectalis muscle, which maintains the anorectal angle that provides fecal continence, relaxes, straightening the anorectal canal. This internal sphincter relaxation and rectal contraction would produce defecation unless the external anal sphincter, composed of striated, voluntarily controlled muscle, contracts in a squeezing reflex, driven by pudendal nerve reflexes, through the sacral spinal cord. This voluntary contraction of the external sphincter buys time, typically 1 to 2 minutes, for the social and positional arrangements that safe defecation requires, before the voluntary inhibition is released and defecation occurs. The defecation posture, squatting rather than sitting, which straightens the anorectal angle, is relevant here. The anorectal angle in the squatting position is more obtuse, reducing the resistance to fecal passage and requiring less straining. The anatomical argument for squatting as the natural defecation posture, made through studies comparing colonic transit time and straining effort in squatting versus sitting subjects, has generated both academic and commercial interest in raised toilet footrests as a means of approximating the squatting angle. The defecation reflex is the digestive system's final act, the completion of the transformation that began at the moment of eating. The Helicobacter pylori bacterium, a spiral-shaped gram-negative organism that colonizes the gastric mucosa of approximately half the world's human population, survives the extreme acidity of the stomach by producing urease, which generates ammonia from urea to neutralize the local acid. In its discovery by Barry Marshall and Robin Warren, who shared the Nobel Prize in 2005, overturned the century-old dogma that the stomach was sterile. The story of Helicobacter pylori is one of the most dramatic in 20th century medicine, a story of 2 Australian pathologists who proposed, in 1984, that most peptic ulcers were caused, not by stress and acid, as the entire medical establishment believed, but by a bacterium, and who were met with such skepticism that Barry Marshall chose to infect himself with the organism to prove the point, developing gastritis within days, and then curing himself with antibiotics. Within a decade, the medical world had been transformed. Peptic ulcer disease, formerly a chronic, relapsing condition, managed with acid suppression and lifestyle modification for life, became a curable infection, treatable with a 1 to 2 week course of antibiotics. H. pylori colonizes the gastric antrumen body, surviving in the hostile acid environment through the urease enzyme that hydrolyzes the small amounts of urea. Pylori disrupts the balance between acid production and mucosal defense. Antral colonization stimulates gastrin release that drives acid hypersecretion, while the inflammatory products of the infection impair the mucous bicarbonate barrier. H. pylori is also a class I carcinogen for gastric cancer, the third leading cause of cancer death worldwide, and its eradication reduces the risk of gastric cancer in infected populations. The cephalic phase of digestion, the neural and hormonal responses triggered by the sight, smell, thought, or taste of food before food enters the stomach, accounts for approximately 30% of the total gastric acid secretion of a meal, and demonstrates that the digestive system begins preparing for food before it arrives. The cephalic phase of digestion is the digestive system's anticipatory response, the biological preparation that begins the moment food is perceived, rather than the moment it arrives. The sight of an appetizing meal, its smell, the act of chewing, even thinking about food that one looks forward to eating, all of these activate the vagus nerve, which carries efferent signals from the dorsal motor nucleus of the medulla oblongata to the stomach, stimulating the parietal cells to secrete hydrochloric acid, the chief cells to secrete pepsinogen, and the mucus cells to secrete protective mucus. The vagal stimulation also releases acetylcholine in the gastric wall, which directly stimulates the gastric glands and stimulates the gastrin-releasing peptide from the enteric nervous system, which triggers the G cells of the antrum to release gastrin, the primary hormonal stimulator of gastric acid secretion. The cephalic phase secretion accounts for approximately 25 to 30% of the total gastric acid response to a meal. The gastric phase, triggered by the arrival and distension of food in the stomach, for approximately 50 to 60%, and the intestinal phase for the remainder. The physiological logic of the cephalic phase is clear. By the time food arrives in the stomach, the acid and enzymes are already present and waiting, ready to begin digestion, without the delay that would occur if secretion had to be stimulated from scratch by the arriving food. The well-known experience of salivation and the associated sensation of hunger and anticipation at the smell of food being cooked is the most accessible expression of the cephalic phase in everyday life. The pancreatic ductal cells, the cells lining the ducts of the exocrine pancreas, produce the bicarbonate-rich component of pancreatic juice through the activity of the cystic fibrosis transmembrane conductance regulator protein on their apical membrane, and their loss of function in cystic fibrosis explains why this disease produces progressive pancreatic insufficiency. The CFTR protein, whose mutation produces cystic fibrosis when both alleles are non-functional, is expressed on the apical membrane of the pancreatic ductal cells, where it functions as a chloride channel and also facilitates bicarbonate secretion through interactions with other transporters. When CFTR is absent or dysfunctional, the pancreatic ductal cells cannot secrete the watery, bicarbonate-rich fluid that normally flushes the zymogen-containing secretions from the acini through the ducts and into the duodenum. The result is that the viscous, protein-rich acinar secretions accumulate in the ducts, blocking them and triggering the chronic pancreatitis that progressively destroys the exocrine parenchyma. By adulthood, most patients with classic cystic fibrosis have developed complete pancreatic exocrine insufficiency. They lack the lipase, proteases, and amylase needed for digestion, and require pancreatic enzyme replacement therapy with each meal to prevent steatorrhea and malnutrition. The same CFTR dysfunction also affects the endocrine pancreas over time, with progressive fibrosis eventually impairing the islets of Langerhans and producing cystic fibrosis-related diabetes in approximately 30 to 40% of adult patients. CFTR modulator therapies, particularly the triple combination elexacaftor, tezacaftor, and ivacaftor, have transformed the outlook for bilirubin is the yellow-orange pigment of bile, the pigment that gives bruises their characteristic color progression, and the pigment whose accumulation in the skin and sclerae produces the jaundice that is one of medicine's most visible signs of hepatic or biliary disease. Its production begins when the macrophages of the spleen and liver phagocytose the aged red blood cells at the end of their approximately 120-day lifespan, degrading the hemoglobin and converting the heme ring first to biliverdin, green, and then to bilirubin, yellow-orange, through hemoxygenase and biliverdin reductase. This unconjugated bilirubin is fat-soluble and travels in the blood bound to albumin. In the liver, the hepatocytes take up the unconjugated bilirubin through the OATP transporters, conjugate it with glucuronic acid through UDP glucuronosyltransferase, and secrete the water-soluble conjugated bilirubin into the bile. The clinical distinction between the three types of jaundice depends on where in this pathway the problem lies. Prehepatic jaundice, from excessive hemolysis producing more bilirubin than the liver can conjugate, is characterized by elevated unconjugated bilirubin and dark urine from urobilinogen. Hepatic jaundice, from hepatocellular damage impairing conjugation and secretion, is characterized by elevated mixed bilirubin and pale stools. Posthepatic, or obstructive jaundice, from bile duct obstruction, preventing bilirubin excretion into the bowel, is characterized by conjugated hyperbilirubinemia, very pale stools, no bile pigment reaching the gut, and dark urine from bilirubin excreted by the kidney. The gastric acid-secreting parietal cells are regulated by three distinct pathways: histamine acting on H2 receptors, acetylcholine acting on M3 receptors, and gastrin acting on CCK2 receptors, all of which converge on the intracellular signaling mechanisms that drive the hydrogen-potassium ATPase to the apical membrane, explaining why blocking any one pathway only partially reduces acid secretion. The three-pathway convergent regulation of gastric acid secretion is a physiological design that ensures the stomach can respond to the diverse stimuli of the cephalic, gastric, and intestinal phases of digestion, while also explaining the pharmacology of acid suppression therapy. Histamine, released by the enterochromaffin-like cells of the gastric mucosa, acts on H2 receptors on the parietal cell to increase cyclic AMP, activating protein kinase A, and ultimately driving the HKAT pathway to the apical canalicular membrane. Acetylcholine, from the vagal efferent fibers and the enteric nervous system, acts on M3 receptors to activate phospholipase C, raising intracellular calcium and directly stimulating HKAT pathway activity. Gastrin, released from the G cells of the antrum in response to stomach distension and peptides in the gastric contents, acts on CCK2 receptors to raise intracellular calcium. The three pathways are synergistic. Each potentiates the response to the others, so that the maximal acid output occurs when all three are stimulated simultaneously. The H2 receptor antagonists, cimetidine, ranitidine, famotidine, block the histamine pathway and reduce acid secretion, but the acetylcholine and gastrin pathways remain intact and compensate. The proton pump inhibitors, omeprazole, lansoprazole, pantoprazole, and their successors, irreversibly inhibit the HKAT pathway itself, the final common pathway of all three stimulatory inputs, and therefore produce a more complete and sustained reduction in acid secretion, making them the most effective acid suppression agents available. The regulation of food intake by the digestive system involves a complex hormonal orchestra with both short-term meal-to-meal signals and long-term adiposity signals. Ghrelin from the stomach rises before meals and signals hunger, GLP-1, peptide YY, and cholecystokinin from the intestine rise after meals and signal satiety, and leptin from adipose tissue and insulin from the pancreas provide the longer-term signals of energy store adequacy. The digestive system is not simply a processing organ. It is a major endocrine communicator that continuously reports the body's nutritional state to the brain, influencing appetite, metabolism, and energy balance through a system of remarkable sophistication. Ghrelin, produced by the oxyntic cells of the gastric fundus, is the only circulating hormone that GLP-1, glucagon-like peptide 1, is released from the L cells of the ileum and colon in response to nutrients, particularly fat and protein, reaching the distal intestine, and it simultaneously stimulates insulin secretion from the pancreatic beta cells, suppresses glucagon, slows gastric emptying, and acts on the hypothalamus to suppress appetite. The magnitude of the GLP-1 response is one reason why the ileum and colon are important. They respond to the nutrients that escaped absorption in the proximal intestine, providing a feedback signal proportional to the malabsorption that would otherwise go uncompensated. Peptide YY, released from the same L cells, similarly reduces appetite and slows gastric emptying, contributing to the ileal brake that prevents the distal small intestine from being overwhelmed. The pharmacological exploitation of this system has been transformative. The GLP-1 receptor agonists developed as treatments for type 2 diabetes, semaglutide, liraglutide, tirzepatide, produced the most effective pharmacological weight loss ever achieved through the same satiety-inducing mechanisms that the intestine uses naturally. The microbiome's contribution to immune development is illustrated by the germ-free mouse, raised in completely sterile conditions, without any microbial colonization, which has profoundly underdeveloped gut-associated lymphoid tissue, reduced circulating immunoglobulin levels, an immature immune system that overreacts to stimuli, and multiple systemic immune abnormalities that are largely corrected by introducing a normal microbiome. The germ-free mouse model, developed in the 1940s and 1950s by James Raniers at the University of Notre Dame, remains one of the most powerful tools for understanding the microbiome's role in host physiology, because it allows researchers to study the host in the complete absence of microbes, and then examine what changes when specific microbes or microbial communities are reintroduced. Germ-free mice have dramatically underdeveloped Peyer's patches and mesenteric lymph nodes, the gut-associated lymphoid tissue that normally develops in response to microbial stimulation in early life. Their serum immunoglobulin levels, particularly IgA, are very low. Their regulatory T-cell populations are deficient, and their immune responses are dysregulated toward hyperreactivity. They are more susceptible to certain types of autoimmunity and allergic inflammation than microbiome-colonized mice. Their intestinal epithelial cells have shorter microvilli and produce less mucus. Their metabolic phenotype is abnormal. They are resistant to diet-induced obesity, have altered glucose metabolism, and have different fat deposition patterns. When germ-free mice are colonized with the microbiome from conventionally raised mice, the transfer of the entire microbial community, most of these abnormalities are corrected, with the immune system maturing normally and the metabolic parameters normalizing. The germ-free mouse demonstrates that the microbiome is not optional for the host. It is an integral component of normal development and physiology, without which the host is developmentally incomplete. Before we go any further, come back to the body for a moment. Feel where you are. The weight of the body against whatever surface is receiving it. The particular warmth of the abdomen, where so much of what we have been discussing is quietly continuing. The peristalsis, the absorption, the fermentation of the day's last meal by the microbiome, the liver doing its patient work. You do not need to feel any of this, but it is there. It has always been there. Let the body rest in the knowledge of its own quiet industry. The digestion continues, whether you are aware of it or not. Tonight, for just a moment, you can be aware. And then you can let it go and let the body carry on. The stomach's ability to expand from its empty volume of approximately 75 milliliters to over 1 liter without significantly raising the intragastric pressure, is due to the phenomenon of accommodation, the vagally mediated relaxation of the fundic smooth muscle that increases the compliance of the stomach, allowing large meals to be accommodated with only a small rise in intraluminal pressure. Accommodation is the physiological mechanism that prevents the stomach from becoming rigid, overpressurized. Accommodation prevents this. It is mediated by a vagovagal reflex. The arrival of food in the stomach activates vagal afferent signals that return to the brainstem and are relayed back down vagal efferent fibers to the fundus, where they activate non-adrenergic, non-cholinergic inhibitory neurons that release nitric oxide onto the fundic smooth muscle, causing it to relax and allowing the fundus to expand. This relaxation, which begins even during the act of swallowing, before food has arrived, driven by the cephalic phase, reduces the intragastric pressure despite the arriving food volume, maintaining the pressure at approximately the pre-meal level. Accommodation is impaired by vagotomy, the surgical cutting of the vagus nerve, used historically to treat peptic ulcer disease, and by certain medications including opioids and anticholinergic drugs that interfere with the vagal pathway. Impaired accommodation produces early satiety, the sensation of being full after only a small amount of food, and is a feature of functional dyspepsia and of the post-operative dumping syndrome that follows gastric surgery. The feeling of comfortable fullness after a satisfying meal, rather than pressure and discomfort, is the experiential expression of accommodation working as it should. The enteroendocrine cells, the endocrine cells scattered throughout the intestinal epithelium, constituting approximately 1% of the intestinal epithelial cells, collectively form the body's largest endocrine organ by number of cells, and produce more than 20 distinct hormones that regulate digestion, satiety, insulin secretion, gut motility, and the gut-brain axis. The enteroendocrine cells, also called the diffuse enteroendocrine system, are individually invisible in the intestinal landscape, scattered singly among the millions of absorptive enterocytes and goblet cells of the intestinal epithelium, but collectively they represent a secretory organ of extraordinary hormonal output. Their diversity is remarkable. The I cells produce cholecystokinin, the S cells produce secretin, the K cells produce GIP, the L cells produce GLP-1, GLP-2, peptide YY, and oxyntomodulin. The G cells of the antrum produce gastrin, the D cells produce somatostatin, the enterochromaffin cells produce serotonin, the EC cells produce motilin, and many more. Each cell type is distributed in specific regions of the gut that match the functional logic of its secretion. The I cells are concentrated in the duodenum and proximal jejunum, where fat and protein first arrive. The L cells are concentrated in the ileum and colon, where the distal intestinal contents trigger the ileal brake. The G cells are in the gastric antrum, where luminal peptides trigger gastrin release for acid secretion. The enteroendocrine cells have microvilli on their luminal surface, the open-type cells, that allow them to sample the chemical composition of the intestinal lumen directly, detecting specific nutrients, pH, osmolarity, and bacterial metabolites through receptor proteins on the microvilli, and calibrating their hormone secretion to the luminal composition. These cells are, in the most literal sense, tasting the intestinal contents and translating that taste into hormonal signals that coordinate the entire digestive response. The colon's bacterial community ferments approximately 30 to 40 grams of dietary fiber per day in people consuming adequate plant-based foods, producing approximately 500 to 600 millimoles of short-chain fatty acids, primarily butyrate, propionate, and acetate, that collectively lower the luminal pH, inhibit the growth of pathogenic bacteria, fuel the colonocytes, and regulate host metabolism. The fermentation of dietary fiber by the colonic microbiome is one of the digestive system's most consequential biological events, a metabolic transformation that converts the indigestible remnants of a plant-based diet into bioactive molecules with effects that extend far beyond the colon. The dietary fibers, the non-starch polysaccharides including cellulose, hemicellulose, pectin, and inulin, the resistant starches that escape small intestinal digestion, the oligosaccharides including fructooligosaccharides and galactooligosaccharides, arrive in the colon intact, providing the primary fermentable substrate for the resident. These acids lower the luminal pH to approximately 5.5 to 6 in the proximal colon, an acidification that inhibits the growth of potential pathogens, including Escherichia coli and Clostridium difficile, that are less acid tolerant than the commensal bacteria. Butyrate is absorbed by the colonocytes and oxidized as their primary fuel. Propionate is transported to the liver via the portal blood, where it inhibits cholesterol synthesis and gluconeogenesis. Acetate enters the systemic circulation, crosses the blood-brain barrier, and suppresses appetite through hypothalamic signaling. The short-chain fatty acid production of the colonic fermentation is thus simultaneously a local energy source, a local antimicrobial system, a systemic metabolic regulator, and a central appetite signal. The portal hypertension that develops when liver cirrhosis impairs hepatic blood flow produces the collateral venous channels, the esophageal and gastric varices, the rectal varices, and the caput medusae of the abdominal wall that divert blood around the liver and carry the risk of catastrophic bleeding when the varices rupture. Portal hypertension, the elevation of pressure in the portal venous system above the normal 8 to 12 millimeters of mercury, is the most dangerous hemodynamic consequence of liver cirrhosis, the end-stage fibrosis that follows chronic liver injury from alcohol, viral hepatitis, non-alcoholic fatty liver disease, or other causes. The architecture of the cirrhotic liver is profoundly disrupted. The normal hepatic lobular structure is replaced by nodules of regenerating hepatocytes, separated by bands of fibrous scar tissue, and the distorted architecture of these fibrotic septa and regenerative nodules impedes the flow of blood through the hepatic sinusoids, increasing the resistance to portal blood flow and raising the portal venous pressure. Blood-seeking alternative routes from the portal system to the systemic venous circulation develops collateral channels through vessels that connect the portal and systemic venous beds, normally small and low flow, but expanding under the persistent elevated pressure. The esophageal varices, submucosal veins in the lower esophagus, dilated by the portal pressure, are the most dangerous of these collaterals because they are thin-walled, superficial structures, subjected to the shear stress of food passage and the trauma of reflux acid, with a significant risk of rupture and hemorrhage. Variceal bleeding, when it occurs, is among the most dramatic and most dangerous emergencies in gastroenterology. A sudden, often massive gastrointestinal hemorrhage that carries a mortality of approximately 20% per episode. The management involves emergency endoscopic variceal ligation, pharmacological reduction of portal pressure with vasoactive drugs, and the correction of coagulopathy from the failing liver that impairs the normal hemostatic response. The small intestine's handling of protein begins with the pancreatic proteases, trypsin, chymotrypsin, elastase, and the carboxypeptidases that produce a mixture of small peptides and amino acids from the luminal proteins, and continues with the brush border peptidases and intracellular peptidases of the enterocyte that complete the digestion to amino acids for transport into the portal blood. Protein digestion illustrates the principle of staged digestion that characterizes the entire digestive system, each stage breaking down the substrate to a size manageable by the next stage, so that the final absorbed molecules are exactly the right size and form for transport across the epithelium. In the stomach, pepsin begins the denaturation and initial cleavage of dietary proteins into large polypeptides. In the duodenum, trypsin cleaves peptide bonds at the carboxyl site of lysine and arginine residues, producing fragments of various lengths. Chymotrypsin cleaves at the carboxyl side of aromatic amino acids, phenylalanine, tyrosine, tryptophan. Elastase cleaves at the carboxyl side of small neutral amino acids. The carboxypeptidases A and B work from the free carboxyl terminus of polypeptides, removing amino acids one at a time. Together, these enzymes produce a mixture of approximately 40% free amino acids and 60% small oligopeptides of 2 to 8 amino acids in length. The brush border peptidases, enteropeptidase, aminopeptidases, and the portal blood arriving at the liver after a protein-containing meal carries a rich mixture of amino acids that the hepatocytes extract and direct to protein synthesis, urea production, or gluconeogenesis, according to the body's current metabolic needs. The vitamin B12 deficiency that follows total gastrectomy, the surgical removal of the entire stomach, performed for gastric cancer, occurs not only because intrinsic factor is lost, but also because the stomach's acid production, which liberates dietary B12 from food proteins, is absent, illustrating that B12 absorption is a multi-step process, dependent on normal gastric function at more than one level. Total gastrectomy, the removal of the entire stomach, creates a comprehensive deficit in the first phase of vitamin B12 processing that the digestive system relies on. In the normal gastric phase of B12 processing, the stomach contributes in two ways. First, the hydrochloric acid and pepsin of the stomach's acid milieu denature the food proteins to which dietary B12 is bound, releasing free B12 that can then bind to the R binders. Second, the parietal cells produce intrinsic factor, without which the B12 intrinsic factor complex cannot be formed for absorption in the terminal ileum. After total gastrectomy, both of these contributions are absent simultaneously. There is no acid to release B12 from food proteins, and there is no intrinsic factor to carry the free B12 to its absorptive site. The result is inevitable B12 deficiency, usually appearing 1 to 3 years after gastrectomy, as the body's substantial hepatic B12 stores are gradually depleted, unless regular parenteral B12 supplementation is given. The neurological consequences of B12 deficiency: the subacute combined degeneration of the spinal cord, where demyelination of the posterior and lateral columns produces the combination of sensory ataxia from posterior column damage and spastic weakness from lateral column damage, are irreversible if B12 deficiency persists for too long, making the monitoring and supplementation of B12 after gastric surgery one of the most important long-term nutritional considerations in bariatric and oncological surgery. The Roux-en-Y gastric bypass, one of the most commonly performed bariatric surgical procedures, achieves dramatic and durable weight loss, partly through restriction of food intake by the small gastric pouch, but primarily through the profound changes in gut hormones it produces: enhanced GLP-1 and peptide YY release, altered bile acid signaling, and changes in the gut microbiome composition. The bariatric surgery revolution has illuminated, more clearly than any other intervention, the degree to which the gut, rather than simply the brain, governs food intake, body weight, and metabolic health. The Roux-en-Y gastric bypass creates a small gastric pouch of approximately 30 milliliters, bypasses the rest of the stomach in the duodenum, and connects the pouch directly to a roux limb of jejunum. The bypass means that the ingested food passes from the pouch directly to the jejunum without exposure to the gastric acid, bile, and pancreatic enzymes that normally process it in the duodenum. This anatomical rearrangement produces dramatic changes in gut hormone secretion. The rapid delivery of partially digested food to the distal small intestine produces an exaggerated L-cell response, with GLP-1 and peptide YY rising much higher after meals than before surgery, producing powerful satiety signals and glucose-stimulated insulin secretion. The bypass also profoundly changes The gut microbiome composition. Within weeks of surgery, the microbiome shifts toward a pattern associated with leanness, and these microbiome changes have been shown to contribute independently to the metabolic improvements of the surgery. Bile acids, whose composition and circulating levels change substantially after gastric bypass, act as metabolic hormones through the FXR and TGR5 receptors, contributing to improved glucose metabolism and increased energy expenditure. The metabolic improvements following gastric bypass, including the dramatic remission of type 2 diabetes that often occurs within days of surgery before significant weight loss, cannot be explained by caloric restriction alone, and point to the gut's hormonal and microbial systems as primary drivers of metabolic health. Zinc. Zinc absorption illustrates the general principle of feedback-regulated mineral absorption that characterizes many of the essential trace elements. The intestinal absorption of zinc depends on the family of ZIP, ZRT/IRT-like protein, and ZNT, zinc transporter, transporters in the enterocytes, ZIP4 on the apical membrane, mediates zinc uptake from the lumen, and ZNT1 on the basolateral membrane, mediates zinc export into the portal blood. When intracellular zinc is high, indicating adequate body stores, zinc induces the expression of metallothionein, a cysteine-rich protein with high affinity for zinc that sequesters zinc within the enterocyte, preventing its export to the portal blood and increasing its loss when the enterocyte is shed into the lumen at the end of its 3 to 5 day lifespan. This metallothionein-mediated retention and loss is the feedback mechanism. High zinc stores produce high intracellular metallothionein, which captures zinc and prevents its absorption, reducing net absorption. Low zinc stores produce low metallothionein, maximizing absorption. Phytate, the phosphate storage molecule of plant seeds, abundant in whole grains, legumes, and nuts, forms insoluble complexes with zinc that cannot be transported by the zinc transporters, substantially reducing the bioavailability of zinc from plant sources. This is why vegetarians and vegans have higher zinc requirements than omnivores, and why populations with heavily plant-based diets in developing countries have high rates of zinc deficiency, despite adequate total zinc content in the food supply. The intestinal epithelial cells are replaced every 3 to 5 days, one of the fastest cell turnover rates in the body, through the continuous proliferation of the stem cells at the base of the intestinal crypts, driven by the WNT signaling pathway and the escalator-like movement of the daughter cells up the crypt-villus axis as they differentiate into the absorptive enterocytes, goblet cells, and enteroendocrine cells of the villus. The intestinal epithelium's 3 to 5 day renewal cycle, the fastest of any epithelial tissue in the body, is both a remarkable biological achievement and a striking vulnerability. The crypts of Lieberkahn, the glandular invaginations between the villi, contain at their base approximately 4 to 6 intestinal stem cells, maintained in their stem cell state, by WNT signals from the Paneth cells and the mesenchymal cells of the crypt base. The LGR5 positive intestinal stem cells divide approximately every 24 hours, producing daughter cells that begin to differentiate as they move upward along the crypt-villus axis. The differentiation trajectory is governed by a series of signaling gradients. WNT signaling is highest at the crypt base, promoting stemness, and notch signaling determines whether a cell differentiates toward the absorptive lineage, enterocyte, or the secretory lineage, goblet cell, Paneth cell, enteroendocrine cell. As the cells migrate upward along the villus, they mature, the enterocytes developing their microvilli, acquiring their digestive enzyme expression, and gaining their transport proteins, until they reach the villus tip, where they are shed into the intestinal lumen by a process that involves both programmed cell death and the mechanical shear forces of luminal flow. The entire villus epithelium is replaced in 3 to 5 days. This rapid renewal is essential for recovery from intestinal injury. The small intestinal epithelium can regenerate substantially within days after significant damage, but it also creates a vulnerability. Any condition that disrupts the stem cells, such as radiation damage, the toxic effects of chemotherapy, or the infiltration of the crypt base by pathogens, can produce the extensive epithelial damage that causes the mucositis of chemotherapy and radiation. The vagus nerve's role in the gut-brain axis is primarily afferent. Approximately 80 to 90% of the fibers in the vagus carry signals from the gut to the brain, rather than from the brain to the gut, making the gut one of the major sensory organs, reporting to the brain about the state of the internal environment. The vagus nerve, named for its wandering course from the brainstem through the neck and thorax, the visceral sensory neurons, whose axons form the afferent vagus, have their cell bodies in the nodose ganglion, and they respond to mechanical stimulation of the gut wall, the stretch of the stomach during a meal, the peristaltic contractions of the intestine, to chemical stimuli in the lumen, sampled via the enteroendocrine cells, and to inflammatory and immunological signals from the gut immune system. These signals project to the nucleus tractus solitarius in the medulla, which distributes the information to the hypothalamus, the limbic system, and the cortex. The gut's influence on mood, stress, and cognitive function, the phenomena summarized by the term gut-brain axis, are largely mediated by these ascending vagal signals. The serotonin produced by the enterochromaffin cells of the gut acts on the vagal afferent terminals to generate signals that reach the brain and influence mood. The microbial metabolites of the gut microbiome can similarly act on the vagal afferents, and the post-meal signals of satiety, comfort, and well-being are primarily vagally transmitted feelings of the gut's satisfied state. The gut is not simply the digestive organ. It is one of the body's primary sensing organs, continuously reporting to the brain. The exocrine pancreas demonstrates a remarkable degree of reserve capacity. Approximately 90% of pancreatic exocrine function must be lost before malabsorption of fat and protein becomes clinically apparent, a reserve that explains why chronic pancreatitis produces symptomatic pancreatic insufficiency only in its advanced stages. The pancreatic exocrine reserve, the enormous overcapacity that the pancreas maintains for enzyme production, is one of the digestive system's most striking examples of biological redundancy. The normal exocrine pancreas produces approximately 10 to 20 times the amount of digestive enzymes required for complete digestion of a typical meal. Even when significant pancreatic tissue has been destroyed by repeated bouts of pancreatitis, by chronic alcohol damage, or by the progressive fibrosis of cystic fibrosis, the remaining functional tissue can produce enough enzymes to maintain adequate digestion, as long as approximately 10% of the original capacity remains. Only when the destruction exceeds approximately 90% does the enzyme production fall below the threshold required for complete digestion, producing the steatorrhea, fat malabsorption with fatty, floating, foul-smelling stools, and the protein malabsorption that are the clinical hallmarks of pancreatic exocrine insufficiency. This reserve also explains why pancreatic cancer, which often obstructs the pancreatic duct and destroys pancreatic parenchyma, typically presents late, with significant pancreatic insufficiency, weight loss, and advanced disease, rather than early, with the easily detectable symptom of malabsorption. The reserve capacity that protects normal digestion for so long also conceals the development of the cancer until it has reached an advanced stage. The approximately 90% destruction threshold before clinical symptoms appear is both a protection for daily function and an obstacle to early cancer detection. The colonic microbiome communicates with the host immune system through pattern recognition receptors, particularly the toll-like receptors and nod-like receptors of the intestinal epithelial cells and lamina propria immune cells that detect the molecular patterns of bacteria and trigger calibrated immune responses that maintain the balance between tolerance of commensals and defense against pathogens. The intestinal immune system faces a challenge that is, in immunological terms, unique. It must simultaneously maintain complete tolerance to the trillions of commensal bacteria that populate the gut lumen and the mucosa, organisms that express the same molecular patterns that would trigger vigorous inflammatory responses in any other tissue, while remaining fully capable of mounting effective defensive responses against the pathogenic bacteria that periodically breach the gut barrier. The resolution of this apparent paradox involves multiple, overlapping mechanisms. The toll-like receptors, the pattern recognition receptors that detect the lipopolysaccharide of gram-negative bacteria, the peptidoglycan of gram-positive bacteria, and the flagellin of modal bacteria, TLR4, which detects LPS, is expressed on the basolateral rather than the apical surface of intestinal epithelial cells. So it only encounters LPS that has translocated through the epithelium, a signal that something has gone wrong with the barrier. rather than the LPS that is normally confined to the lumen. Regulatory T cells in the lamina propria, conditioned by the microbiome's metabolites, including butyrate and the short-chain fatty acids, actively suppress the inflammatory responses that would otherwise be triggered by bacterial antigen sampling. The result is a state of controlled, low-grade immunological awareness, a continuous immunological conversation with the microbiome that maintains the mucosal immune readiness without the tissue-damaging inflammation that would make the gut dysfunctional. The concept of the gut microbiome-brain axis, the bidirectional communication between the gut microbial community and the central nervous system through neural, hormonal, and immunological pathways, has generated extraordinary research interest after findings linking gut microbiome composition to mood disorders, anxiety, autism spectrum disorder, and neurodegenerative disease. The gut-brain axis has moved from a curiosity at the margins of neuroscience to a central research area in just over a decade, driven by a series of striking findings in both animal models and human cohort studies. In animal studies, germ-free mice show altered anxiety and stress responses, abnormal social behavior, and dysregulated hypothalamic pituitary adrenal axis responses to stress, abnormalities that can be partially corrected by introducing specific microbial species, particularly Lactobacillus rhamnosus, and related organisms that reduce anxiety-like behavior when given as probiotics. In humans, studies have found correlations between gut microbiome composition and major depression, with depressed patients showing reduced lactobacillus and bifidobacterium and elevated potentially inflammatory bacteria. The most dramatic human evidence came from a 2019 study that found bidirectional causality, transferring the fecal microbiome from depressed patients to germ-free rats produced depressive behaviors in the rats that were absent when the rats received microbiome from non-depressed donors. The pathways of communication are multiple. The vagal afferents from the gut transmit signals of the gut's microbial state to the brainstem and higher brain regions. The microbial production of neurotransmitter precursors, most of the body's serotonin, is made from tryptophan by gut bacteria and enterochromaffin cells, and gut bacteria produce GABA, dopamine precursors, and numerous other neuroactive metabolites, influences the systemic availability of these molecules. The microbiome's immune modulation influences the neuroinflammation that is increasingly recognized as a feature of psychiatric and neurodegenerative disorders. The mechanisms are being characterized, but the central finding that the microbiome influences the brain now commands serious scientific attention. The liver's role in immune defense through the Kupffer cells that line the hepatic sinusoids and phagocytose bacteria, endotoxin, and cellular debris arriving in the portal blood makes it the body's primary filter for the bacteria that continuously translocate in small amounts across the healthy intestinal barrier, protecting the systemic circulation from portal bacteremia. The liver's immunological function is as important as its metabolic function and is perhaps less appreciated. The portal blood arriving from the intestine contains, even in health, small amounts of bacteria and bacterial products, lipopolysaccharide, peptidoglycan, bacterial DNA, that have translocated across the intestinal epithelial barrier through the normal but small permeability of the healthy gut. This translocation is a normal and continuous process, not a sign of pathology, and the liver's Kupffer cells, the specialized macrophages, that line the hepatic sinusoids and constitute approximately 30 to 40% of all macrophages in the body, are positioned precisely to intercept these translocation products before they can reach the systemic circulation. The Kupffer cells express pattern recognition receptors, toll-like receptors and scavenger receptors that recognize. The Kupffer cells also play a role in the immune activation and inflammatory response when bacterial translocation increases, as it does in liver disease, where the gut barrier is compromised by portal hypertension and the gut microbiome dysbiosis of cirrhosis, and where the increased bacterial translocation drives the systemic inflammation and the risk of spontaneous bacterial peritonitis that are features of advanced liver disease. The liver is thus not only the metabolic guardian of the portal blood, but its immunological guardian, the last checkpoint between the microbiome-laden portal circulation and the systemic blood. The taste sensation of umami, produced by the glutamate receptors of the taste cells in the taste buds of the tongue and palate, is not merely a sensory experience, but triggers specific cephalic phase responses, including salivary and gastric secretion, that prepare the digestive system for the protein-rich meal that glutamate-containing foods typically predict. Umami, the fifth taste, alongside sweet, salty, sour, and bitter, was identified as a distinct taste quality by Japanese chemist Kakuni Ikeda in 1908, who isolated glutamate from kombu seaweed as the active molecule responsible for the distinctive savoriness of the broth. The taste cells expressing the metabotropic glutamate receptor mGluR4 and the T1R1T1R3 heterodimer, the taste receptor for umami, detect the free glutamate present in protein-rich foods, including meat, fish, aged cheeses, tomatoes, mushrooms, and fermented products. The perception of umami, of savoriness, of the deep, lingering quality that distinguishes a rich broth from water, is the taste system's signal that protein is present in the food, and it triggers a set of cephalic phase responses, specifically calibrated to prepare for protein digestion, an increase in salivary protease production, a vagally mediated increase in gastric acid secretion and pepsinogen release, and an increase in pancreatic enzyme secretion. The cephalic phase response to umami is measurably distinct from the responses to sweet or salty tastes. It specifically stimulates the protein-digesting preparatory responses, rather than the carbohydrate-digesting ones, triggered by sweetness. Glutamate has also been found to act not only on the taste buds, but on glutamate receptors in the intestinal epithelium, where it stimulates enteroendocrine cell secretion, including CCK, which coordinates the protein digestive response, establishing a second glutamate sensing point, downstream of the initial taste. Umami is, in this sense, the taste of protein, the signal that sets the digestive system's protein processing program in motion from its first moment of detection. The process of gluconeogenesis, the synthesis of glucose from non-carbohydrate precursors, including amino acids, lactate, and glycerol, primarily occurs in the liver, and to a lesser extent the kidney, and represents the digestive metabolic system's response to fasting, ensuring continuous glucose supply to the brain and red blood cells, even when no carbohydrate is being absorbed from the gut. Gluconeogenesis is the liver's commitment to the brain, the biochemical process by which the liver generates the glucose that the brain requires continuously, regardless of whether carbohydrate is currently being absorbed from the gut. During fasting in between meals, when intestinal glucose absorption has ceased, the liver first mobilizes its glycogen stores, releasing glucose into the bloodstream. When glycogen stores are depleted, approximately 12 to 24 hours into a fast, the liver switches to gluconeogenesis, synthesizing new glucose from three main precursors. Lactate, produced by red blood cells and by working muscle, is recycled to glucose through the Cori cycle, which regenerates the glucose that was consumed in glycolysis, while shifting the metabolic burden from the periphery to the liver. Amino acids, particularly alanine, which is produced by muscle proteolysis and transamination, and which carries the amino groups of other amino acids to the liver in a form that allows their carbon skeletons to enter the gluconeogenic pathway, provide the substrate for approximately 60% of gluconeogenic glucose production during prolonged fasting. Glycerol, released from the hydrolysis of triglycerides in adipose tissue, provides the remaining substrate. The liver's gluconeogenic capacity is so essential that its failure, in fulminant hepatic failure, rapidly produces hypoglycemia that must be treated with intravenous glucose infusion. The intestinal tight junctions, the protein complexes that seal the paracellular space between adjacent intestinal epithelial cells and determine the permeability of the intestinal epithelial barrier, are regulated dynamically by signals from the immune system, the microbiome, dietary components, and inflammatory mediators, making intestinal permeability a physiologically variable property rather than a fixed anatomical feature. The tight junction is the intestinal barrier's fundamental seal, the molecular gasket between adjacent epithelial cells that prevents the unregulated passage of luminal contents, bacteria, toxins, food antigens, through the paracellular space into the lamina propria. The tight junction is not a single protein, but a complex assembly of transmembrane proteins, the claudins, occludin, and the junction adhesion molecules, connected to the actin cytoskeleton of the epithelial cells by scaffolding proteins including ZO1, ZO2, and ZO3. The claudins are particularly important. Different claudin isoforms form either barrier-forming or pore-forming strands, and the specific composition of claudins in the tight junction determines its permeability to ions and small molecules. This composition can be dynamically regulated. Myosin light chain kinase, activated by inflammatory cytokines, including tumor necrosis factor and interferon gamma, phosphorylates myosin, contracting the parajunctional actomyosin ring and pulling the tight junction proteins apart, increasing permeability. Butyrate from the microbiome stabilizes tight junctions by increasing claudin expression and reducing myosin light chain kinase activity. Dietary factors including zinc, vitamin D, and quercetin similarly support tight junction integrity, while gliadin, the protein of wheat that produces the immune response of celiac disease, activates zonulin release from intestinal epithelial cells, and zonulin directly opens tight junctions by triggering the actomyosin contraction that separates the junctional proteins. The dynamic regulation of intestinal permeability, its increase in inflammation, stress, and dysbiosis, and its restoration by dietary fiber, a healthy microbiome, and specific micronutrients, makes it one of the most important and most modifiable features of gut health. The gallbladder's concentration of bile, a 10-fold reduction in the volume of hepatic bile, is driven by the active absorption of sodium and chloride by the gallbladder epithelium, with water following osmotically, and the failure of this absorption when the gallbladder mucosa is inflamed, cholecystitis, impairs the concentration of bile and the emulsification function it provides. The gallbladder epithelium is a specialized absorptive epithelium that works in the opposite direction to the usual intestinal epithelium. Rather than absorbing nutrients from the lumen, it absorbs water and electrolytes to concentrate a secretion. The gallbladder's tall columnar cells express the NACAT passe on their basolateral membrane, maintaining the sodium gradient that drives the sodium absorption through apical sodium transporters, with chloride following through the CFTR chloride channel, relevant in cystic fibrosis, where gallbladder disease is a common associated problem. Water follows osmotically, reducing the volume of bile by up to 90% during the fasting period between meals, when bile is stored in the gallbladder. The rate of concentration depends on the transit time. The longer bile remains in the gallbladder, the more concentrated it becomes, and is increased by fasting and reduced by eating. Cholecystitis, inflammation of the gallbladder, usually from gallstone impaction in the cystic duct, disrupts the epithelial function and impairs bile concentration, changing the rheological properties of the gallbladder contents and further promoting stone formation. The gallbladder's concentration function is also the reason that fasting before cholecystography, the imaging of the gallbladder, is essential. Fasting allows the gallbladder to fill and concentrate bile for 6 to 8 hours before imaging, making the stones or other pathology visible. The gallbladder after a meal, having expelled its concentrated bile, in response to cholecystokinin, is small and empty, and poorly suited for imaging. The parietal cells of the stomach secrete not only hydrochloric acid, but also intrinsic factor in the proton pump, the hydrogen-potassium ADPase. From the same cell, the parietal cell's structural specialization for its secretory function is among the most dramatic examples of cellular architecture serving cellular function in the body. In the resting state, the parietal cell contains an extensive intracellular network of smooth membrane vesicles, the tubulovesicles, studded with the hydrogen-potassium ADPase pump, stored in this intracellular compartment, away from the apical surface. When the parietal cell is stimulated by histamine, acetylcholine, or gastrin, the tubulovesicles fuse with the apical membrane of the intracellular canaliculus, the deep invagination of the apical surface that gives the parietal cell its characteristic appearance, dramatically increasing the surface area of the secretory membrane and increasing the density of the HKAT passe at the secretory surface up to tenfold. This membrane recruitment is the mechanism by which the parietal cell can rapidly amplify its acid-secreting capacity. In response to a meal, the pumps are already synthesized and packaged. They simply need to be moved to the active surface. The proton pump inhibitors, the most widely prescribed drugs in the world, work by irreversibly binding to the HKAT passe on this canalicular membrane, blocking its enzymatic activity and suppressing acid secretion until new pumps are synthesized, taking approximately 3 to 5 days for full restoration of secretory capacity. The parietal cell's ability to turn its secretory capacity up and down by recruiting or internalizing the membrane containing the HKAT passe, rather than by synthesizing and degrading the pumps themselves, is one of the fastest and most energy-efficient ways of controlling a secretory rate in human biology. The concept of dysbiosis, the disruption of the normal gut microbiome composition and function, has been associated with an expanding range of conditions, including inflammatory bowel disease, colorectal cancer, obesity, type 2 diabetes, cardiovascular disease, non-alcoholic fatty liver disease, and psychiatric disorders, suggesting that maintaining microbiome health may be one of the most important determinants of overall health. Dysbiosis is, in a sense, the microbiome's equivalent of the inflammatory response, a shift from the normal, balanced microbial community toward a state that is less able to perform the microbiome's health-promoting functions and more likely to drive pathological processes. The term covers several distinct types of microbiome disruption: loss of diversity, reduced species richness and evenness, which impairs the microbiome's functional redundancy and resilience, overgrowth of potentially pathogenic species, reduction in the beneficial short-chain fatty acid producers, and disruption of the spatial organization of the microbiome, including the breach of the inner mucus layer by organisms that should be confined to the outer layer. Each of these disruptions has been associated with specific pathological consequences, many of which are mediated through the short-chain fatty acid production, the tight junction regulation, and the immune modulation that the healthy microbiome normally performs. The association between dysbiosis and inflammatory bowel disease is the most thoroughly studied. Patients with Crohn's disease and ulcerative colitis consistently show reduced Faecalibacterium prosnitzii, the major butyrate producer, and increased proteobacteria, including adherent invasive E. coli strains that colonize the mucosa and drive the inflammatory response. Whether the dysbiosis causes the inflammation or the inflammation causes the dysbiosis remains a subject of active research. Most likely both directions of causality are relevant, creating a self-reinforcing cycle of microbiome disruption and intestinal inflammation. The translational implication that restoring a healthy microbiome through dietary fiber, fermented foods, and targeted probiotic or prebiotic interventions might prevent or treat these conditions is one of the most exciting and most contested hypotheses in current medicine. There are so many places left to go in the digestive system tonight. The extraordinary biology of the colon's bacterial fermentation, the liver's relationship with fat and alcohol, the gut's role in immune tolerance, the connection between what you eat and who you are at a cellular level. If you would like to be here for those journeys, and for all the other quiet wonders that Relax with Science brings to the night, one slow evening at a time, subscribing keeps the door open between us. The absorption of calcium in the small intestine, primarily in the duodenum and proximal jejunum, is regulated by the active form of vitamin D, 1,25-dihydroxyvitamin D3, or calcitriol, which increases the expression of the calcium-binding protein calbindin and the apical calcium channel TRPV6, and is enhanced by the acidic pH of the proximal intestine that keeps calcium in its soluble ionized form. Calcium absorption is one of the most carefully regulated nutrient absorption processes in the body, reflecting the critical importance of calcium for bone health, neuromuscular function, and intracellular signaling, and the potential toxicity of calcium excess if regulation fails. Approximately 30% of dietary calcium is absorbed under normal conditions, but this fraction can rise to as high as 60% in states of high demand, growth, pregnancy, lactation, and calcium deficiency, when parathyroid hormone stimulates the renal hydroxylation of 25-hydroxyvitamin D to calcitriol, which in turn increases intestinal calcium absorption. Calcitriol acts on the nuclear vitamin D receptor of the enterocyte to increase the transcription of TRPV6, the apical calcium channel that allows calcium entry from the lumen, and of calbindin D9K, the intracellular calcium binding protein that shuttles calcium from the apical entry channel to the basolateral calcium ATPase that exports it into the portal blood. The dependence of calcium absorption on the luminal pH explains why acid suppression therapy with proton pump inhibitors can reduce calcium absorption by eliminating the gastric acid that normally dissolves calcium salts in the stomach and maintains the low pH of the proximal duodenum, where calcium is most efficiently absorbed. Long-term proton pump inhibitor use has been associated with reduced bone mineral density and increased fracture risk in multiple epidemiological studies, likely at least partly through this impairment of calcium absorption, though the absolute risk increase is modest. The concept of the gut as the body's second brain, coined by gastroenterologist Michael Gershon in his 1998 book of the same name, was initially controversial, but is now supported by an extensive body of evidence showing that the enteric nervous system can generate complex coordinated behaviors without any input from the central nervous system, and that 90% of the serotonin in the body is in the gut. Michael Gershon's claim that the gut deserves to be considered a second brain was provocative in 1998, when the enteric nervous system was known, but its independence and complexity were not widely appreciated in medicine or in public discourse. The evidence that has accumulated since then has largely vindicated the concept. The enteric nervous system's neuron count, approximately 500 million, equaling or exceeding the spinal cord, and its organizational complexity, with sensory neurons, interneurons, motor neurons, and glial cells, all using the neurotransmitters of the central nervous system, provides the anatomical basis for a system capable of autonomous operation. The physiological demonstration that gut peristalsis, secretory responses, and blood flow regulation can all be coordinated by the enteric nervous system after complete surgical denervation from the central nervous system provides the functional basis. The serotonin statistic, that approximately 90 to 95% of the body's total serotonin is synthesized and stored in the gut, is among the most striking, because serotonin is so strongly associated in the public mind with brain function and mood regulation. The gut's serotonin, produced by the enterochromaffin cells and released onto the vagal afferents and the enteric neurons, acts as the primary trigger for the peristaltic reflex and as a signal to the brainstem about the state of the gut, a visceral serotonin system that operates in parallel with the brain's synaptic serotonin system. The two systems are linked. Drugs that increase serotonin in the brain, the selective serotonin reuptake inhibitors, also alter the gut serotonin system, which is one reason why gastrointestinal symptoms, including nausea, diarrhea, and altered bowel, the appendix, that 3 to 10 centimeter vermiform structure, attached to the cecum at the beginning of the large intestine, is among the most famous supposedly vestigial organs in human anatomy, invoked in discussions of evolutionary relics as evidence of our distant past. The reassessment of the appendix's function began with the observation that its walls are rich in lymphoid tissue. The Peyer's patches of the appendix are particularly dense relative to its size, making it part of the gut-associated lymphoid tissue system. Its biofilm-protected lumen, opening at the cecum through a narrow orifice that protects it from being flushed during diarrheal illnesses, gives it the structural characteristics of a refuge for beneficial bacteria. Randall Bollinger and colleagues proposed the safe house hypothesis in 2007. The appendix acts as a reservoir of the commensal gut bacteria, protected from the clearing effects of diarrheal illness that can recolonize the colon after the infectious diarrhea has resolved. Studies in populations with and without appendices have found higher rates of Clostridioides difficile recurrence after appendectomy, consistent with the loss of the bacterial reservoir that would normally outcompete C. difficile during the repopulation phase. The appendix's lymphoid tissue also makes it a site of early immune activation for gut antigens. While the loss of an appendix does not produce obvious long-term immune or microbiome deficiencies, in most people, the redundancy of the gut immune system is substantial. The organ is neither functionless nor truly vestigial, and its evolutionary persistence in many mammalian lineages reflects a real, if modest, biological function. The role of bile acids as hormones, acting through the farnesoid X receptor in the liver and intestine, and the G-protein-coupled receptor TGR5 in the intestine, brown adipose tissue, and gallbladder, makes them regulators of glucose and lipid metabolism, thermogenesis, and energy expenditure, extending their function far beyond their classical role in fat emulsification. Bile acids were for most of medical history understood simply as detergent molecules, amphipathic compounds that emulsified dietary fat for digestion and were recycled efficiently through the enterohepatic circulation. The discovery in the 1990s and 2000s that bile acids are ligands for nuclear receptors and G-protein-coupled receptors transformed this picture, establishing bile acids as systemic hormones with metabolic functions that go far beyond the intestine. The farnesoid X receptor, FXR, is activated by bile acids in the hepatocyte and the ileal enterocyte, and when activated, it suppresses bile acid synthesis in the liver. Negative feedback increases bile acid transport in the ileum and regulates lipid and glucose metabolism. In the ileum, FXR activation stimulates the secretion of fibroblast growth factor 19, FGF19, which travels via the portal blood to the liver, suppressing bile acid synthesis and stimulating glycogen synthesis. FXR agonism reduces hepatic lipogenesis, improves insulin sensitivity, and is the target of obeticholic acid, a bile acid derivative used in the treatment of primary biliary cholangitis and being investigated in non-alcoholic steatohepatitis. TGR5, the G-protein-coupled bile acid receptor, when activated in the intestinal L cells, stimulates GLP-1 secretion, connecting bile acid signaling directly to the incretin axis. In brown adipose tissue, TGR5 activation increases intracellular thyroid hormone activity, promoting thermogenesis and energy expenditure. The altered bile acid signaling after Roux-en-Y gastric bypass, with higher postprandial bile acid levels reaching the distal intestine, is now recognized as one of the mechanisms by which bariatric surgery improves metabolic health beyond simple caloric restriction. The Paneth cell, the specialized secretory cell at the base of the intestinal crypt, that produces the antimicrobial peptides defensins, lysozyme, and secretory phospholipase A2, is the guardian of the crypt stem cell niche, protecting the intestinal stem cells from microbial invasion and providing the WNT and Notch signals that maintain stem cell identity and drive their proliferation. The Paneth cell occupies one of the most strategically important positions in the intestinal epithelium, at the very base of the crypt, interdigitated among the intestinal stem cells, where it performs two distinct but related functions: microbial defense and stem cell support. Its antimicrobial arsenal is formidable. The alpha defensins, cryptins, are small, positively charged peptides that insert into bacterial membranes, forming pores that disrupt secretory phospholipase A2 degrades bacterial phospholipids. Together, these secretions create a sterile zone around the intestinal stem cells, ensuring that the crypts, where the progenitor cells that renew the entire intestinal epithelium reside, are not colonized by the bacteria that inhabit the rest of the intestinal lumen. Simultaneously, the Paneth cells are the primary source of the WNT3 and EGF signals that maintain the LGR5 positive stem cells in their undifferentiated, proliferative state at the crypt base. Without Paneth cell WNT signaling, the intestinal stem cells lose their stemness and differentiate prematurely. The disruption of Paneth cell function by the NOD2 mutations associated with Crohn's disease, by autophagy defects, or by ER stress from misfolded protein accumulation, reduces both the antimicrobial defense of the crypt and the maintenance of the stem cell niche, potentially contributing to the epithelial barrier dysfunction and the inadequate mucosal regeneration that characterize Crohn's disease. The non-alcoholic fatty liver disease spectrum, from simple hepatic steatosis through non-alcoholic steatohepatitis to cirrhosis, has become the most common liver disease in high-income countries, affecting approximately 25 to 30% of the global adult population, and its pathogenesis involves the interplay of dietary excess, insulin resistance, gut microbiome dysbiosis, and hepatic lipotoxicity. Non-alcoholic fatty liver disease, the accumulation of fat in the liver in the absence of significant alcohol consumption, has followed the epidemiological trajectory of the obesity epidemic, growing from a relatively rare condition to the most prevalent liver disease on Earth within the span of 3 decades. The fat accumulates in the hepatocytes when the delivery of fatty acids to the liver from dietary fat, from de novo lipogenesis driven by dietary carbohydrate, and from the lipolysis of adipose tissue, in the context of insulin resistance, exceeds the liver's capacity to oxidize fat or to export it as VLDL particles. Simple steatosis, fat accumulation without inflammation or fibrosis, is largely benign. But in a subset of patients, the fat accumulation triggers oxidative stress, endoplasmic reticulum stress, and mitochondrial dysfunction that activate the inflammatory response and drive the hepatocyte death and fibrosis of non-alcoholic steatohepatitis. The gut microbiome contributes to this progression. Dysbiosis in NASH is associated with increased intestinal permeability, increased translocation of bacterial products, particularly LPS from gram-negative bacteria, into the portal blood, and activation of the hepatic toll-like receptors that drive the inflammatory response. Short-chain fatty acid production is reduced in NASH, reducing butyrate's anti-inflammatory effects on the gut epithelium. And the microbiome's production of ethanol, a little appreciated fact, that healthy gut bacteria produce small amounts of ethanol through fermentation, is elevated in NASH patients, contributing to hepatic lipid accumulation through the same mechanism as dietary alcohol. NASH has become the leading indication for liver transplantation in many countries, a stark measure of the medical burden of a condition driven substantially by the dietary and microbiome changes of the modern food environment. The digestive system's extensive involvement in immune function through the gut-associated lymphoid tissue, the secretory IgA system, the epithelial barrier, the microbiome, and the tolerogenic mechanisms of the mesenteric lymph nodes makes the gut the site of the majority of the body's immune activity, a fact reflected in the statistic that approximately 70 to 80% of the immune cells in the body reside in the gut. The gut is, in immunological terms, the body's most active and most challenged immune site. No other organ faces the dual challenge of the intestine to absorb the nutrients of food without triggering immune responses to the food antigens that inevitably accompany those nutrients, while simultaneously maintaining full defensive capability against the pathogens that use the same alimentary route as food. The immunological infrastructure of these cells, the plasma cells producing secretory IgA, are perhaps the most numerous. The gut contains more IgA-secreting plasma cells than all other tissues combined, producing the 3 to 5 grams of secretory IgA secreted into the gut lumen each day. The Peyer's patches and mesenteric lymph nodes are the sites of the oral tolerance induction that prevents the immune system from mounting destructive responses to food antigens. A failure of this tolerance is what produces the celiac disease response to gluten and the food allergies that affect a growing proportion of the population. The gut's immunological importance is not confined to the gut itself. The immune cells educated in the gut-associated lymphoid tissue circulate through the body, influencing systemic immunity. The cytokines produced in gut inflammation reach the systemic circulation, and the microbiome's influence on immune development shapes the immune system's responses to allergens, infections, and autoantigens throughout life. A second question. I wonder how the body is feeling right now. Whether the day has settled into something quieter, whether the digestive system's quiet work of the evening has contributed to the heaviness of the limbs and the softening of the thoughts. Whether the breath has slowed. If any of that is true, even slightly, I am glad. And if you feel like leaving something in the comments about where you are in the world tonight, or what the evening has felt like, I would genuinely love to read it. sometimes the most meaningful thing is simply, I was here. The night was kind. That is enough. The fecal microbiota transplant, the transfer of stool from a healthy donor into the gut of a recipient, is now an established treatment for recurrent Clostridioides difficile infection, with cure rates exceeding 90% that are dramatically superior to antibiotics alone, demonstrating that restoring microbiome ecology can cure infectious disease where antibiotics have failed. Fecal microbiota transplantation is one of medicine's more conceptually radical innovations. The idea that introducing another person's gut bacteria could cure a potentially fatal infection has a visceral quality that made it difficult to take seriously before the clinical trial evidence made it impossible to ignore. Clostridioides difficile, C. diff, is a spore-forming gram-positive anaerobe that colonizes the gut when the normal microbiome is disrupted by antibiotics, typically during hospitalization. It produces toxins A and B that damage the colonic epithelium, causing pseudomembranous colitis, the profuse watery diarrhea, abdominal pain, and systemic inflammation that can be fatal in elderly or immunocompromised patients. Antibiotic treatment with vancomycin or fidaxomicin kills C. diff, but also further disrupts the microbiome, leaving the gut vulnerable to recurrence in approximately 25 to 30% of patients, and each recurrence increases the probability of the next. FMT works by restoring a diverse, healthy microbial community that outcompetes C. diff through colonization resistance, the combination of competition for nutrients, production of inhibitory short-chain fatty acids and bacteriocins, and restoration of the intact mucus layer, and the secondary bile acid metabolism that inhibits C. diff germination. The standard protocol, a single infusion of donor stool via colonoscopy into the cecum, achieves approximately 90 to 95% cure rates in multiply recurrent C. diff, with the remaining cases cured by a second infusion. The success of FMT for C. diff has catalyzed research into its application for other conditions associated with dysbiosis, with ongoing trials in inflammatory bowel disease, graft versus host disease, and metabolic syndrome. The enteric nervous system develops from neural crest cells, the migratory cells that also give rise to the peripheral ganglia, the adrenal medulla, and the melanocytes, and its colonization of the gastrointestinal tract, from mouth to anus, occurs during weeks 5 to 12 of fetal development, with failure of complete colonization, producing Hirschsprung's disease. The embryological development of the enteric nervous system is one of the more remarkable examples of directed cellular migration in developmental biology. Neural crest cells from the vagal region of the neural crest, at the level of the somites adjacent to rhombomeres 6 to 8 of the hindbrain, migrate into the foregut at approximately embryonic week 5, and then progressively colonize the developing gut, from the esophagus through to the rectum. Over the next this caudal colonization front The wave of migrating neural crest cells that moves from mouth toward anus is driven by the receptor tyrosine kinase RET and its ligand GDNF, expressed on the neural crest cells and the gut mesenchyme respectively. The RET-GDNF signaling acts as both a survival signal and a chemotactic signal, guiding the neural crest cells along the gut wall and stimulating their proliferation. If this migration fails to reach the colon, as occurs in Hirschsprung's disease, caused by mutations in RET or its coreceptors, the affected colonic segment lacks enteric ganglia, is aganglionic, cannot generate peristalsis, and remains in a state of tonic contraction. The result is a functional obstruction that presents in the newborn period as failure to pass meconium and abdominal distension, requiring surgical resection of the aganglionic segment. The length of the affected segment varies. Approximately 70% of cases involve only the rectosigmoid, short segment Hirschsprungs, while approximately 5% involve the entire colon, total colonic aganglionosis, and very rare cases involve the entire gut. The condition's genetics, with multiple susceptibility loci including RET, EDNRB, and several others, reflect the complexity of the developmental program that normally ensures the complete colonization of the gut by enteric neural crest cells. The concept of the food matrix, the physical and chemical structure of whole foods, that determines the rate and extent of nutrient digestion and absorption, explains why the metabolic response to eating almonds differs dramatically from the response to eating almond butter or almond flour, even when the macronutrient composition is identical. The food matrix is one of nutritional science's most important and least appreciated concepts. The recognition that nutrients do not exist as isolated molecules in food, but are embedded in a complex physical structure of cells, cell walls, fibers, proteins, and lipids that profoundly influences how they are digested and absorbed. The almond example is instructive. Intact almonds contain roughly the same total fat, protein, and carbohydrate as almond butter or almond flour, but the fat in intact almonds is stored in oil bodies within intact cell walls that resist complete digestion by human enzymes and mechanical processing during chewing. Studies have shown that the human gut absorbs approximately 20% fewer calories from whole almonds than from equivalent almond preparations where the cell walls have been disrupted by grinding. The cell walls that encase the almond's fat effectively serve as a physical barrier to lipase, reducing the rate and extent of fat digestion. The metabolic consequences of this matrix effect are significant. Intact whole almonds produce a lower postprandial insulin response, lower triglyceride rise, and higher satiety than equivalent almond flour, despite identical macronutrient content. The same matrix effect applies across the food supply. The glycemic index of whole grain pasta is lower than white flour pasta not only because of the fiber, but because the pasta structure resists rapid starch gelatinization. The intact cell walls of legumes slow protein digestion compared to equivalent protein isolates. The food matrix is the reason why whole foods and ultra-processed foods with identical macronutrient profiles can have dramatically different metabolic effects. The structure of the food is as metabolically important as the chemical composition. The absorption of fat-soluble vitamins A, D, E, and K is linked to fat absorption and shares the chylomicron pathway, explaining why very low-fat diets, conditions of fat malabsorption, and drugs that interfere with fat absorption can all produce deficiencies of these vitamins, even when dietary intake is adequate. The fat-soluble vitamins are incorporated into the mixed micelles, the bile salt lipid assemblies that carry the products of fat digestion to the brush border for absorption and enter the enterocyte by passive diffusion or by specific transporters at the same time as the monoglycerides and fatty acids that form the body of the micelle. Once inside the enterocyte, they are incorporated into the chylomicrons, along with the triglycerides and cholesterol synthesized in the smooth endoplasmic reticulum. Cholestatic liver disease, which reduces bile salt secretion, impairs fat emulsification, and fat-soluble vitamin absorption. Crohn's disease involving the terminal ileum impairs bile salt reabsorption, depletes the bile salt pool, and reduces fat and fat-soluble vitamin absorption. The drug orlistat, which inhibits pancreatic lipase to reduce dietary fat absorption as a weight loss treatment, produces fat-soluble vitamin deficiency as a side effect, requiring supplementation. Very low-fat diets, below approximately 15 grams of fat per day, reduce the micellar phase that carries the vitamins, impairing their absorption even when vitamin intake is adequate. The practical implication is that fat-soluble vitamins are best absorbed with meals that contain some fat, and that supplement forms of these vitamins that are taken with a fat-free meal or drink may be poorly absorbed. The regulation of gastric motility and emptying is impaired in diabetic gastroparesis, the delayed gastric emptying that affects approximately 30 to 40% of people with long-standing type 1 and type 2 diabetes, due to the autonomic neuropathy that damages the vagal and enteric innervation of the stomach, producing nausea, vomiting, and unpredictable glucose absorption that complicates insulin management. Diabetic gastroparesis is one of the most challenging complications of long-standing diabetes because it disrupts the fundamental assumption that underlies insulin therapy, that the rate of food absorption can be predicted from the content and timing of a meal. In normal gastric physiology, the stomach controls the rate of delivery of nutrients to the small intestine with considerable precision, and the insulinotropic hormonal response to nutrients is calibrated to this controlled delivery rate. In gastroparesis, the stomach's motility is impaired. The migrating motor complex may be absent or fragmented, the antral contractions weak, and the pyloric sphincter discoordinated, producing highly variable and unpredictable gastric emptying. A meal that takes 30 minutes to leave the stomach in normal physiology may take 4 to 6 hours, or may sit largely undigested for 24 hours in severe gastroparesis. For a patient on insulin, this variability is dangerous. A pre-meal bolus of insulin calculated for normal gastric emptying produces hypoglycemia when the stomach empties slowly, and then hyperglycemia hours later, when the delayed emptying finally delivers the nutrients to the absorptive small intestine. The pathophysiology of diabetic gastroparesis involves multiple interacting mechanisms: loss of the interstitial cells of Cajal, the pacemaker cells of the gastric smooth muscle, due to chronic hyperglycemia and oxidative stress, damage to the vagal efferents that coordinate gastric motility, and loss of the inhibitory nitrogen neurons of the enteric nervous system that normally ensure appropriate pyloric relaxation. The condition exemplifies the systemic impact of poor glycemic control and the interdependence of the digestive and endocrine systems. The liver's synthesis of clotting factors, including fibrinogen, prothrombin, and factors V, 7, 9, X, 11, and 12, makes it indispensable for hemostasis and the impaired clotting factor production of advanced liver disease produces the coagulopathy that is one of the most dangerous aspects of cirrhosis, contributing to the risk of catastrophic hemorrhage from esophageal varices. The liver's contribution to hemostasis encompasses almost every component of the coagulation cascade: most of the procoagulant factors, most of the anticoagulant proteins, and many of the fibrinolytic proteins are produced exclusively or predominantly by the hepatocytes. The vitamin K-dependent clotting factors, prothrombin, factor 2, factor 7, factor 9, factor X, protein C, protein S, and protein Z, require the hepatic enzyme gamma-glutamyl carboxylase to add the carboxyl groups to their glutamate residues that enable them to bind calcium and participate in the coagulation cascade. Warfarin's anticoagulant effect is through the inhibition of this vitamin K-dependent carboxylation, reducing the activity of these factors. In liver disease, the synthesis of all clotting factors is progressively impaired as the functional hepatocyte mass is reduced by fibrosis and necrosis. The prothrombin time, which measures the function of the extrinsic pathway and the common pathway, is prolonged, reflecting the gut microbiome's dual character, highly responsive to short-term dietary perturbations but also maintaining a relatively stable long-term composition, is one of the more nuanced findings of the microbiome research era. David et al.'s 2014 study in Cell demonstrated that when healthy volunteers were switched from their habitual diet to either a completely animal product diet or a completely plant-based diet, the microbiome composition changed substantially within 2 to 3 days. The animal-based diet increased the bile-tolerant organisms including Bilophila wadsworthia and bacteroids and reduced the fiber-fermenting firmicutes, while the plant-based diet increased the fiber-fermenting organisms. But when the volunteers returned to their habitual diet, the microbiome largely returned to its baseline composition within a few days, demonstrating the stability of the long-term core microbiome. The individual's gut microbiome has a characteristic composition, a signature, that is more similar between identical twins than between fraternal twins, reflecting a genetic component and is more similar between individuals sharing a household than between unrelated individuals sharing the same geographic region, reflecting the role of shared environment and transmission. Early life exposures. Mode of delivery: vaginal versus cesarean section, breastfeeding versus formula feeding, antibiotic exposure in infancy. Shape the microbiome in ways that persist for years, and the first 3 years of life appear to be a critical window during which the microbiome composition has lasting effects on immune development, allergy risk, and metabolic trajectories. The practical implication is that short-term dietary changes can shift the microbiome, but sustained dietary patterns over months and years produce the more durable changes in microbiome composition that are associated with the long-term health effects of dietary patterns. The stomach's secretion of approximately 2 liters of gastric juice per day, containing hydrochloric acid, pepsinogen, intrinsic factor, and mucus, is matched by an equal and opposite secretion of bicarbonate into the portal blood during acid production, the so-called alkaline tide, that briefly raises the pH of the venous blood after a meal. The alkaline tide is a physiological curiosity that directly demonstrates the chemistry of acid production in the stomach. Every hydrogen ion pumped into the gastric lumen by the parietal cell is generated from carbonic acid, water plus carbon dioxide, catalyzed by carbonic anhydrase, which simultaneously produces a bicarbonate ion. This bicarbonate ion is exported across the basolateral membrane of the parietal cell into the portal blood in exchange for a chloride ion. The result is that for every mole of hydrochloric acid secreted into the stomach, an equal mole of sodium bicarbonate is secreted into the portal blood. The alkaline tide that slightly raises the pH of the portal blood after a meal and contributes to the mild post-meal respiratory alkalosis that can be detected in highly sensitive blood gas measurements. The scale of this effect is not trivial. When the stomach is maximally stimulated, it can secrete approximately 30 millimoles of acid per hour, generating an equivalent 30 millimoles of bicarbonate per hour into the portal blood. The kidney responds to this alkaline tide by excreting more bicarbonate in the urine, the post-meal alkaline urine that Pavlov first described, as a compensatory adjustment that returns the systemic acid-base balance to normal. The alkaline tide is also the physiological basis of the metabolic alkalosis that develops in severe, prolonged vomiting. When the stomach's acid secretions are repeatedly emptied by vomiting before the bicarbonate they generate can be reabsorbed from the duodenum, the bicarbonate remains in the blood and the potassium depletion and volume contraction of vomiting prevent the kidney from excreting it, producing the hypokalemic, hypochloremic metabolic alkalosis of the bulimic patient or the pyloric stenosis infant. The liver's metabolism of alcohol, primarily through the alcohol dehydrogenase pathway in the cytosol and the microsomal ethanol oxidizing system in the smooth endoplasmic reticulum, produces acetaldehyde as an intermediate, and it is this acetaldehyde, rather than alcohol itself, that is responsible for most of the cellular toxicity, DNA damage, and inflammatory signaling that underlies alcoholic liver disease. Alcohol metabolism in the liver follows two main pathways, each with different kinetics and producing the same toxic intermediate. The primary pathway, alcohol dehydrogenase, using NAD+ as the cofactor, converts ethanol to acetaldehyde in the cytosol, producing NADH in the process. The excess NADH generated by heavy alcohol metabolism shifts the hepatocyte's redox state dramatically toward reduction, inhibiting gluconeogenesis, causing hypoglycemia, inhibiting fatty acid oxidation, causing fat accumulation, alcoholic steatosis, and inhibiting the tricarboxylic acid cycle, shifting metabolism away from oxidative phosphorylation. The secondary pathway, the microsomal ethanol oxidizing system, using cytochrome P452 × 10⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻⁻ ethanol to acetaldehyde in the endoplasmic reticulum, generating reactive oxygen species as a byproduct. Acetaldehyde, the common product of both pathways, is a highly reactive molecule that forms adducts with proteins, disrupts mitochondrial function, inhibits DNA repair, activates stellate cells to produce collagen, and stimulates Kupffer cell production of TNF-alpha and IL-8. It is the proximate mediator of the inflammation and fibrosis of alcoholic hepatitis and alcoholic cirrhosis. The enzyme acetaldehyde dehydrogenase converts acetaldehyde to the relatively harmless acetate in the mitochondria, the genetic variants in this enzyme that produce acetaldehyde accumulation in some Asian populations, causing the flushing, nausea, and tachycardia of the alcohol flush reaction, demonstrate the toxicity of acetaldehyde when it is not rapidly metabolized. The concept of the migrating motor complex in the fasting state, the 90-minute cycles of intestinal housekeeping that prevent bacterial overgrowth, is disrupted by the postprandial state, and the timing of meals therefore has direct consequences for bacterial populations in the small intestine, with frequent snacking potentially promoting the bacterial overgrowth that contributes to bloating and irritable bowel symptoms. The timing of meals and the intervals between them are not simply a matter of social convention or hunger management, but have direct consequences for the microbial ecology of the small intestine. In the fasting state, the migrating motor complex cycles every approximately 90 minutes, propelling the bacteria and debris of the small intestinal lumen aborally toward the colon and maintaining the relatively sparse bacterial population of the healthy small intestine. Each feeding event, including small snacks, interrupts the MMC and replaces the fasting motor pattern with the fed motor pattern, which is designed for digestion and absorption rather than for cleansing. If meals and snacks are consumed so frequently that the fasting state is never re-established for the 1 to 2 hours needed for the MMC to complete a full cycle, the small intestine never undergoes the housekeeping sweeps that prevent bacterial accumulation. The practical consequence of this continual feeding pattern, common in modern grazing eating habits, is a slow drift toward higher bacterial populations in the small intestine, with the potential for symptom development in susceptible individuals. The association between frequent snacking and small intestinal bacterial overgrowth is not firmly established in randomized controlled trials, but the mechanistic plausibility is clear. and the clinical observations from gastroenterologists treating SIBO suggest that extended fasting intervals between meals, allowing the MMC to complete multiple cycles, can reduce bacterial counts and symptoms. This is one of the physiological rationales for the practice of time-restricted eating, extending the overnight fast to ensure adequate MMC housekeeping cycles, although the evidence base remains evolving. The celiac disease immune response to gluten, the proteins of wheat, rye, and barley, is a T-cell-mediated adaptive immune response to gluten peptides presented on the HLA-DQ2 and HLA-DQ8 molecules by antigen-presenting cells in the small intestinal mucosa, producing villus atrophy and malabsorption in genetically predisposed individuals. Celiac disease is one of the most thoroughly characterized examples of a food-triggered autoimmune condition, a condition in which the immune system's adaptive response to a dietary protein drives tissue destruction in the organ that absorbs it. Gluten, the storage protein complex of wheat, comprising the gliadin and glutenin fractions, is partially resistant to digestion by the intestinal brush border peptidases, leaving long peptides in the small intestinal lumen that can penetrate the intestinal epithelium through both paracellular and transcellular routes. Once in the lamina propria, certain gliadin peptides are modified by the enzyme transglutaminase 2, which deamidates specific glutamine residues to glutamate, increasing their charge, and the modified peptides have dramatically increased affinity for the HLA-DQ2 or HLA-DQ8 molecules expressed on the antigen-presenting cells of genetically susceptible individuals. Approximately 95% of celiac disease patients carry HLA-DQ2 or HLA-DQ8. The HLA peptide complexes are presented to CD4 plus T cells in the lamina propria, triggering an immune response that includes T helper cell activation, B cell activation producing anti-transglutaminase 2 antibodies, the diagnostic serological marker, and cytotoxic T cell activation that kills the enterocytes. The villus atrophy that results, the destruction of the villi that provides the enormous absorptive surface of the small intestine, produces malabsorption of virtually all nutrients, including iron, calcium, fat-soluble vitamins, and folate. The discovery that transglutaminase 2 modification of gluten peptides is required for efficient immune recognition explains why HLA-DQ2/DQ8 possession alone, present in approximately 30% of the population, is not sufficient to cause celiac disease, and why other genetic and environmental factors, including the composition of the gut microbiome, influence disease development. The production of digestive enzymes is regulated not only by meal-triggered hormonal signals but by the phenomenon of enzyme induction, the increase in the synthesis of specific digestive enzymes in response to dietary loads of their substrates over days to weeks, allowing the digestive capacity to adapt to the habitual dietary composition. The regulation of digestive enzyme production operates on two timescales. The short-term, meal-by-meal regulation, the CCK-stimulated pancreatic enzyme secretion and the secretin-stimulated bicarbonate secretion described earlier, ensures that the appropriate enzyme mix is delivered to the duodenum for each meal's specific nutrient content. The long-term adaptation, enzyme induction over days to weeks, adjusts the total synthetic capacity of the digestive system to match the habitual dietary composition. People consuming high-protein diets chronically upregulate the synthesis and secretion of pancreatic proteases, trypsinogen, chymotrypsinogen, so that their maximum proteolytic capacity exceeds that of people on lower protein diets. People consuming high-starch diets upregulate pancreatic amylase synthesis. The brush border lactase expression, as discussed earlier, represents the most extreme and best studied example of dietary enzyme induction and its evolutionary regulation. This adaptive enzyme induction reflects the transcriptional regulation of the digestive enzyme genes by substrate load. The substrate arriving in the intestine generating signals through the enteroendocrine cells and the enteric nervous system that reach the pancreatic acinar cells in the intestinal epithelium and alter their gene expression. The practical implication is that a person who dramatically changes their diet, moving from a high carbohydrate to a very high fat diet, for instance, may initially experience digestive discomfort as the enzyme mix does not match the new dietary composition, but over 2 to 4 weeks the enzyme production adapts to the new diet, and symptoms typically resolve. The gut takes time to learn a new diet. The splanchnic circulation, the blood supply to the digestive organs including the stomach, small intestine, large intestine, liver, spleen, and pancreas, distributed through the celiac artery, the superior mesenteric artery, and the inferior mesenteric artery, receives approximately 30% of the cardiac output at rest and is a major reservoir of blood volume that can be mobilized by sympathetic vasoconstriction during exercise or hemorrhage. The splanchnic circulation is one of the cardiovascular system's largest and most variable regional circulations, a network of vessels serving the most metabolically diverse collection of organs in the body, whose collective blood flow requirements vary enormously between the fasting and postprandial states. At rest in the fasting state, the splanchnic organs receive approximately 1,500 milliliters per minute, approximately 25 to 30% of the cardiac output. After a meal, the postprandial hyperemia, the increase in intestinal blood flow driven by the metabolic activity of digestion and absorption and by the local release of vasoactive substances including substance P, nitric oxide, and vasoactive intestinal peptide from the enteric nervous system, increases splanchnic blood flow by approximately 50 to 100%, directing the cardiovascular system's resources toward the organs doing the work of nutrient processing. This postprandial increase in splanchnic blood flow at the expense of other vascular beds explains the feeling of lethargy after a large meal. The blood redistribution reduces cerebral and musculoskeletal perfusion, and explains why vigorous exercise immediately after a large meal can produce ischemic abdominal pain in some individuals, as the exercise-related sympathetic vasoconstriction of the splanchnic bed competes with the postprandial need for increased intestinal flow. In hemorrhage and in intense exercise, The sympathetic nervous system's splanchnic vasoconstriction mobilizes the blood volume sequestered in the large, distensible splanchnic veins back into the central circulation, providing an autotransfusion effect that helps maintain cardiac output. The gut microbiome's production of secondary bile acids. The chemical transformation of primary bile acids secreted by the liver into the secondary bile acids deoxycholic acid and lithocholic acid by colonic bacteria creates molecules with different receptor specificities and different metabolic effects, connecting the liver's bile acid synthesis to the colonic microbiome's biotransformation capacity. The secondary bile acid pathway is one of the most metabolically consequential examples of host microbiome co-metabolism, a biosynthetic collaboration in which the liver performs the first chemical steps and the colonic bacteria perform the final transformations, producing molecules that act as hormones throughout the body. The primary bile acids, cholic acid and chenodeoxycholic acid, are synthesized in the liver from cholesterol through a multi-step enzymatic pathway. They are conjugated with taurine or glycine in the hepatocyte before secretion into bile, forming the water-soluble bile salts that emulsify dietary fat. In the colon, where approximately 5% of the primary bile acids escape ileal reabsorption and reach the bacterial fermentation zone, the gut bacteria, primarily Clostridium scindens and related 7-alpha dehydroxylating bacteria, remove the 7-alpha hydroxyl group that distinguishes the primary from the secondary bile acids, converting cholic acid to deoxycholic acid and chenodeoxycholic acid to lithocholic acid. Deoxycholic acid, the most abundant secondary bile acid in the human colon, is reabsorbed from the colon, enters the portal blood, and is recycled through the enterohepatic circulation alongside the primary bile acids. It activates FXR and TGR5 receptors with different relative affinities from the primary bile acids, contributing a distinct hormonal signal to the bile acid pool. Elevated deoxycholic acid is associated with colorectal cancer. It is a potent promoter of colonic epithelial cell apoptosis resistance and proliferation, and the gut microbiome composition's influence on the ratio of primary to secondary bile acids is one mechanism by which diet, through its effects on the microbiome, influences colorectal cancer risk. The regulation of gut permeability by the microbiome, through the short-chain fatty acid butyrate stabilization of tight junctions, through secretory IgA's prevention of bacterial mucosal contact, and through the mucus-maintaining role of the mucin-degrading bacteria that signal for mucus renewal, makes microbiome health a primary determinant of intestinal barrier integrity. The intestinal barrier is not a static, fixed structure maintained by the epithelial cells alone, but a dynamic system continuously maintained and modulated by the microbial community that inhabits the lumen. The microbiome contributes to barrier integrity through multiple mechanisms that operate at different structural levels. At the level of the mucus layer, the outermost physical barrier, the mucin-degrading bacteria including Akkermansia mucinifila, while degrading the mucus, simultaneously stimulate goblet cells to produce replacement mucus, maintaining the mucus layer thickness. When a Muciniphala is at healthy levels. This stimulation-regeneration balance keeps the inner mucus layer intact. At the level of the tight junctions, the molecular seals between epithelial cells, butyrate produced by the fermentative bacteria acts on the butyrate receptor GPR109A and through histone deacetylase inhibition to increase the transcription of claudin-1, occludin, and ZO1, the proteins that constitute the tight junction complex. At the immunological level, the secretory IgA produced in response to the microbiome coats the bacteria and prevents them from approaching the epithelial surface, maintaining the spatial separation between microbiome and epithelium that prevents bacterial overstimulation of the epithelial pattern recognition receptors. The disruption of any of these microbiome-mediated barrier maintenance mechanisms by reduced muciniphala abundance, by reduced butyrate producers, or by reduced IgA production, compromises the intestinal barrier and allows bacterial products to reach the lamina propria, triggering the systemic low-grade inflammation associated with metabolic disease, autoimmunity, and neurological conditions. The development of food allergies, IgE-mediated hypersensitivity reactions to specific food proteins, reflects a failure of the normal oral tolerance that the mucosal immune system develops toward food antigens, and the hygiene hypothesis that reduced early microbial exposure impairs tolerance development provides a framework for understanding the rising prevalence of food allergy in high-income countries. Food allergy has increased in prevalence by approximately 50% in high-income countries over the last 20 to 30 years. A rise too fast to be explained by genetic change and too consistent across countries to be coincidental. The mechanism of food allergy involves a failure of the oral tolerance that the gut immune system normally develops toward food antigens, the process by which the mucosal immune system learns to treat repeatedly encountered food proteins as harmless rather than as threats requiring an immune response. Normal oral tolerance is mediated by the tolerogenic dendritic cells of the mesenteric lymph nodes, which present food antigens to naive T cells in the presence of retinoic acid and TGF-beta, converting them to regulatory T cells that suppress rather than mount immune responses to the same antigens on future encounter. The hygiene hypothesis, and its more specific successor, the old friends hypothesis, proposes that the reduced microbial exposure of children in high-income countries, fewer infections, C-section delivery, formula feeding, reduced contact with soil and animals, antibiotic use, impairs the development of the regulatory immune circuits that oral tolerance requires, tilting the immune system toward Th2 responses including IgA production. The microbiome's role in this process is specific. The production of short-chain fatty acids by the microbiome promotes regulatory T-cell development. Certain bacterial species including Clostridia clusters 4 and XIVA that colonize the intestinal epithelium are particularly potent inducers of regulatory T-cells, and early life depletion of these bacteria by caesarean section, antibiotic exposure, or lack of breastfeeding creates the immunological conditions for allergic sensitization to food proteins. The enteroendocrine L cells of the ileum and colon, the same cells that produce GLP-1 and peptide YY, also produce GLP-2, a glucagon-like peptide that specifically stimulates intestinal epithelial proliferation, enhances nutrient absorption, and increases intestinal blood flow, making it a trophic hormone for the gut epithelium, whose synthetic analog teduglutide is used to treat short bowel syndrome. GLP-2 was identified as a glucagon-like peptide in 1984. The sequence of the proglucagon chain was found to encode not one but two glucagon-like peptides, and GLP-2 is processed from the same L-cell precursor as GLP-1 and released simultaneously into the circulation. Its functional role remained obscure for over a decade until Daniel Drucker's group demonstrated in 1996 that GLP-2 was a specific intestinotrophic factor, a hormone that promoted growth and function specifically in the intestinal epithelium. GLP-2 acts through the GLP-2 receptor on the enteric neurons and indirectly on the epithelial cells to stimulate crypt cell proliferation, reduce enterocyte apoptosis, increase the expression of brush-border digestive enzymes, enhance nutrient transport, and increase intestinal blood flow. Its net effect is to increase the absorptive surface area and function of the intestinal epithelium. In the healthy intestine, GLP-2 contributes to the postprandial response that optimizes absorptive capacity after a meal. In short bowel syndrome, the catastrophic reduction in absorptive surface area that follows the surgical removal of large segments of small intestine, commonly from Crohn's disease, mesenteric ischemia, or surgical complications, the remaining intestinal epithelium must compensate through adaptive hypermutation. It can increase villus height, increase crypt depth, and increase its absorption per unit length. GLP-2 is a primary driver of this compensatory intestinal adaptation, and teduglutide, the GLP-2 receptor agonist approved for short bowel syndrome, accelerates this adaptation, reducing parenteral nutrition requirements in patients who would otherwise be permanently dependent on intravenous nutrition. The timing of food consumption relative to the body's circadian rhythm influences digestion, metabolism, and the gut microbiome composition, with the emerging field of chrononutrition demonstrating that the same meal consumed at breakfast versus dinner produces different glycemic responses, different lipid profiles, and different microbiome responses due to the circadian variation in digestive and metabolic processes. Chrononutrition, the study of the timing of food consumption in relation to the body's internal clocks, has emerged as a significant research area from the intersection of circadian biology and nutritional science. The digestive system has its own circadian rhythm. The gastric emptying rate is faster in the morning than in the evening. The activity of brush-border digestive enzymes peaks in the morning. The postprandial insulin response is greater in the morning than the evening for the same glucose load, reflecting both greater pancreatic beta cell responsiveness and greater peripheral insulin sensitivity. And bile acid secretion varies across the day. The practical consequence of these circadian variations is that the metabolic response to food is not constant across the day. Studies by Sachidananda Panda and others have shown that the same caloric load consumed early in the day produces lower postprandial glucose, lower insulin, and lower triglyceride peaks than the same meal consumed late at night, and that the insulin sensitivity of peripheral tissues including skeletal muscle is substantially higher in the morning than in the evening. These differences are clinically significant. Individuals who eat most of their calories at breakfast compared to dinner lose more weight, have better glycemic control, and have lower cardiovascular risk markers, even when total caloric intake and macronutrient composition are identical. The gut microbiome itself has a circadian rhythm. The relative abundance of different bacterial species oscillates across the day in temporal patterns that are disrupted by eating at night and by shift work, providing another mechanism by which meal timing influences the microbiome-mediated aspects of metabolic health. The hepatic stellate cell, the parasinusital cell of the liver that normally stores vitamin A as retinal esters and cytoplasmic lipid droplets, becomes the primary fibrosis-producing cell of the liver when activated by chronic injury, transforming from its quiescent vitamin A storing state into an activated myofibroblast that deposits collagen and contracts the hepatic sinusoids. The hepatic stellate cell is the key cellular mediator of hepatic fibrosis, the process that converts acute or chronic liver injury into the scarring that ultimately produces cirrhosis. In the healthy liver, the stellate cells lie in the parasinusital space of Dease, the narrow space between the hepatocytes and the endothelium of the hepatic sinusoids, in their quiescent state, characterized by their prominent cytoplasmic lipid droplets that store approximately 80% of the body's total vitamin A as retinal esters. In this quiescent state, the stellate cells perform important regulatory functions including regulation of sinusoidal blood flow through their contractile properties and production of paracrine signals to the hepatocytes. When the liver is injured by alcohol, by viral hepatitis, by fat accumulation in NASH, by cholestatic disease, or by any cause of sustained hepatocellular damage, the activated Kupffer cells and damaged hepatocytes release the stellate cell activating signals, TGF-beta, platelet-derived growth factor, reactive oxygen species, and the damage-associated molecular patterns from injured cells. The stellate cells respond by losing their vitamin A lipid droplets, proliferating, and transdifferentiating into myofibroblasts, the collagen-secreting, contractile cells that deposit the fibrous tissue of hepatic fibrosis. Once activated, the stellate cell-derived myofibroblasts secrete type I and III collagen into the space of Dease, transforming the normal low-resistance hepatic sinusoid into a collagen-lined channel of increased resistance, the basis of portal hypertension. The current hope for reversing cirrhosis, through elimination of the underlying injury combined with antifibrotic therapy, is focused on eliminating the activated stellate cells by inducing their apoptosis or by reversing their activation back to the quiescent state. The concept of food synergy, the enhancement of the biological effect of nutrients by other components of the same food or meal, explains why the health effects of whole dietary patterns typically exceed what would be predicted from the individual nutrients they contain, and is one of the reasons why nutritional supplementation often fails to replicate the benefits of whole food consumption. Food synergy is the nutritional science principle that dismantles the reductionist view of foods as delivery vehicles for specific nutrients, revealing instead that the biological effects of food arise from the interactions among its countless components. The most studied example is the synergy between the fat-soluble vitamins and the dietary fat in the same meal. Vitamin D from fatty fish is absorbed far more efficiently with the fat in that fish than if the same dose of vitamin D was taken in a fat-free supplement with a fat-free meal. Because the fish fat provides the micellar solubilization and the chylomicron packaging that the vitamin requires for absorption. The lycopene of cooked tomatoes, a carotenoid with potential antioxidant and prostate cancer protective effects, is absorbed approximately 3 times more efficiently from tomato sauce with olive oil than from raw tomatoes or from lycopene supplements, because the oil provides the lipid vehicle for micellar incorporation and the heat processing of the tomatoes disrupts the cell walls that otherwise limit lycopene release. The iron from spinach is more efficiently absorbed when spinach is consumed with vitamin C-containing foods, the ascorbic acid reducing the ferric iron to the ferrous form that the intestinal iron transporter can accept. The polyphenol of green tea and turmeric are more bioavailable when consumed with the piperine of black pepper, which inhibits the glucuronidation enzymes that would otherwise metabolize them too rapidly for absorption. These examples reflect a broader biological reality. Plants have evolved complex matrices of interacting molecules that work together, and isolating one molecule and concentrating it in a supplement typically removes it from the synergistic context in which its biological effects were observed. The discovery that the appendix harbors a safe house of commensal bacteria has been extended by the observation that mucus-associated biofilms in the folds of the appendiceal mucosa, protected from the flushing of diarrheal contents, can reseed the colon after recovery, but the biofilm-forming capacity of the appendix may also make it a potential harbor for biofilm-protected pathogenic bacteria that contribute to appendicitis. The appendix's dual potential, as both a protective reservoir for beneficial bacteria and a potential sanctuary for pathogens, is one of the more nuanced aspects of its revised biological role. The mucus-associated biofilm that forms in the irregular folds of the appendiceal mucosa provides physical protection to both beneficial and pathogenic organisms, and the bacteria that colonize these biofilms may differ from the planktonic bacteria that dominate the colonic lumen. In the healthy appendix, the biofilm is dominated by commensal organisms, including Fusobacterium species, Bacteroidetes, and various firmicutes, whose presence provides the reservoir function that allows recolonization after diarrheal illness. In appendicitis, the inflammation of the appendix, affecting approximately 7% of the population at some point in their lifetime, the accumulation of bacteria in the appendiceal lumen combined with obstruction of the narrow appendiceal orifice produces the bacterial overgrowth, mucosal invasion, and subsequent inflammatory cascade that produces the characteristic right lower quadrant pain, fever, and elevated white cell count. Recent studies have identified specific bacterial species, particularly Fusobacterium nucleatum and Escherichia coli, in the mucosa of inflamed appendices, and have proposed that the appendix's biofilm-protected environment allows the accumulation of these organisms to the point of mucosal penetration that initiates the inflammatory process. The appendix's story is thus more complex than either the classic view, purely vestigial, functionless, or the revisionist view, purely protective reservoir. It is an organ whose structural features simultaneously enable its reservoir function and create a vulnerability to the bacterial accumulation that causes appendicitis. The intestinal gas, produced by the bacterial fermentation of undigested carbohydrates in the colon, composed primarily of nitrogen, carbon dioxide, hydrogen, and methane, is responsible for approximately 200 milliliters of gas passage per day in the average adult, and its composition and volume reflect the specific bacterial community and the substrate it receives. Intestinal gas is one of the more socially fraught topics in gastroenterology and one of the more informationally rich. The composition and volume of intestinal gas is a direct readout of the fermentative activity of the gut microbiome and the substrates reaching the colon. The gas is produced almost entirely in the colon. The small intestine has too few bacteria to produce significant gas volumes. By the anaerobic fermentation of the carbohydrates that escape digestion and absorption in the small intestine. The primary substrates are dietary fiber, resistant starch, oligosaccharides including lactose and lactase-deficient individuals, and fermentable oligosaccharides including fructans and galacto-oligosaccharides. Hydrogen gas is produced by the fermentative bacteria as a byproduct of their anaerobic metabolism. It diffuses across the colonic mucosa into the blood and is exhaled in the breath, which is the basis of the hydrogen breath test used to detect carbohydrate malabsorption and small intestinal bacterial overgrowth. Methane is produced by methanogenic archaea, particularly Methanobrevibacter smithii, that use hydrogen to reduce carbon dioxide, converting 4 molecules of hydrogen into 1 molecule of methane and 1 of water and effectively removing hydrogen from the colonic environment. Not all individuals harbor methane-producing archaea, which partly explains the individual variation in intestinal gas composition and in the characteristics of stool. The relative ratio of hydrogen-consuming to hydrogen-producing bacteria and archaea in an individual's microbiome shapes their gas production phenotype, their bowel habit, and their susceptibility to the bloating and distension that can accompany fermentation of high-fiber diets. The protective role of dietary fiber in colorectal cancer, supported by abundant epidemiological evidence showing lower colorectal cancer rates in populations with higher fiber intake, is mediated through multiple mechanisms including dilution of carcinogens by increased stool bulk, reduction of luminal pH by short-chain fatty acid production, enhanced colonic motility reducing contact time between carcinogens in the mucosa, and the direct anti-proliferative effects of butyrate on colonic epithelial cells. The relationship between dietary fiber and colorectal cancer is one of the most consistently replicated findings in nutritional epidemiology, supported by evidence from population studies across diverse dietary patterns, from migrant studies showing that immigrants from low-risk to high-risk countries acquire the colorectal cancer risk of their adopted country within 1 to 2 generations, and from mechanistic studies demonstrating the multiple protective pathways through which fiber acts. Dennis Burkitt's original hypothesis, proposed in the 1970s after his observations of the rarity of colorectal cancer in rural African populations eating high-fiber diets, focused on the dilution and transit time effects. More fiber produced more stool bulk, diluting the carcinogens present in the colonic contents, and increased motility from the bulk reduced the time that carcinogens were in contact with the colonic mucosa. These physical effects remain relevant, but the mechanistic understanding has expanded considerably since Burkitt's time. Butyrate, produced from fiber fermentation by the gut bacteria, acts on colonocytes as a histone deacetylase inhibitor, promoting the expression of genes that induce differentiation and apoptosis in abnormal cells and suppressing the expression of genes that drive proliferation. In cancer cells, where energy metabolism has shifted to anaerobic glycolysis, the Warburg effect, butyrate is not consumed as a fuel but accumulates intracellularly at high concentrations that potently inhibit histone deacetylase activity, producing what is called the butyrate paradox, where the same molecule that feeds normal colonocytes also inhibits cancer cell proliferation. The reduction in luminal pH from short-chain acid production inhibits the conversion of primary to secondary bile acids by reducing the activity of the 7-alpha dehydroxylating bacteria that produce the carcinogenic secondary bile acids deoxycholic acid and lithocholic acid. You have stayed with me a very long time tonight. Through the mouth and the stomach and the small intestine and the liver and the pancreas and the colon and the microbiome and all the extraordinary, patient biology of digestion. 100 facts, and you have been here for nearly all of them. I do not take that lightly. Time is the most honest gift there is. If Relax with Science has earned a small place in your evenings, if these nights have given you something you would like to return to, subscribing is the simplest and most honest way to say so. I will be here. I always will be. Just a little further now. The body has been working all day. We are almost at rest. You have stayed with me a very long time tonight. Through the mouth and the stomach and the small intestine and the liver and the pancreas and the colon and the microbiome and all the extraordinary, patient biology of digestion. 100 facts, and you have been here for nearly all of them. I do not take that lightly. Time is the most honest gift there is. If Relax with Science has earned a small place in your evenings, if these nights have given you something you would like to return to, subscribing is the simplest and most honest way to say so. I will be here. I always will be. Just a little further now. The body has been working all day. We are almost at rest. The ileum's role is the site of bile salt and vitamin B12 absorption, and the disruption of both when the terminal ileum is diseased or resected, makes this final section of the small intestine, despite being structurally similar to the rest of the small intestine, functionally irreplaceable in ways that the jejunum cannot compensate for. The terminal ileum, the last 30 centimeters of the small intestine, is the digestive system's specialist absorber. It handles the molecules that require specific, energy-dependent transport systems to cross the epithelium, rather than the passive or facilitated transport processes that handle the majority of nutrients in the proximal intestine. Bile salts and vitamin B12 are the two physiologically critical substrates that can only be absorbed here, for reasons that illuminate the logic of their unique structural requirements. The bile salt transporter, ASBT, is expressed exclusively in the terminal ileum, providing the active transport mechanism that captures approximately 95% of the bile salts that arrive from the small intestinal lumen before they reach the colon. The cubilin-megalin receptor complex for intrinsic factor B12, as discussed in detail, is similarly restricted to the terminal ileum. These functional specificities mean that Crohn's disease affecting the terminal ileum, by far the most common site of Crohn's disease, simultaneously impairs both bile salt absorption, reducing the bile salt pool and impairing fat digestion, and B12 absorption, producing over time the B12 deficiency that requires parenteral supplementation. The terminal ileum resection performed for Crohn's disease, ischemia, or cancer produces the same dual deficiency, and its management, bile salt replacement or cholestyramine for the bile salt diarrhea, B12 injections, must specifically address both losses. The jejunum, given time, can partially adapt its absorptive capacity for many nutrients after small intestinal resection, but it cannot upregulate the transporter systems for bile salts and B12 that are simply not expressed in jejunal epithelium. The digestive system's immune tolerance to food antigens, the suppression of immune responses to the harmless proteins and carbohydrates of the diet, is actively maintained by the tolerogenic dendritic cells of the mesenteric lymph nodes that convert naive T cells to regulatory T cells in a retinoic acid-dependent process, and its failure produces not only food allergy but also the immune responses to food antigens that drive some of the gut symptoms in inflammatory bowel disease. The maintenance of oral tolerance is not a passive default but an active, continuously maintained immunological program that requires specific cellular populations and molecular signals to be present and functioning. The tolerogenic dendritic cells that present food antigens in the mesenteric lymph nodes are conditioned by the retinoic acid produced by the intestinal epithelium to express the enzyme RALDH2, which metabolizes retinol to retinoic acid, and to produce the tolerogenic cytokine TGF-beta. When these dendritic cells present food antigen peptides to naive T cells in the retinoic acid-rich environment of the mesenteric lymph node, the naive T cells differentiate into FOXP3+ regulatory T cells, the cells that actively suppress immune responses to the same antigens on future encounter. This conversion process is so efficient in the mesenteric lymph nodes that the same dendritic cells that would produce inflammatory responses to the same antigen in a peripheral lymph node produce regulatory responses in the mesenteric environment. The mesenteric lymph node's tolerogenic microenvironment, maintained by the dietary vitamin A that is converted to retinoic acid in the intestinal epithelium, makes oral tolerance fundamentally dependent on adequate vitamin A status, providing a mechanistic explanation for the observation that vitamin A deficiency in developing countries is associated not only with the classic deficiency features of night blindness and immune impairment but also with increased intestinal inflammation and impaired barrier function. The failure of oral tolerance in the context of dysbiosis, barrier disruption, or altered dendritic cell function, when food antigens are presented in inflammatory rather than tolerogenic conditions, is a plausible mechanism for the de novo development of food intolerance in the context of gut disease. The microbiome's influence on drug metabolism, through the bacterial enzymes that can activate prodrugs, inactivate active drugs, or transform drug metabolites back into active or toxic forms, creates significant interpersonal variation in drug response that cannot be predicted from genetic pharmacogenomics alone, and that is beginning to be appreciated as a major source of variable drug efficacy and toxicity. The gut microbiome's metabolic interaction with drugs is one of the newest and most clinically important frontiers in pharmacology. The microbiome expresses a vast repertoire of enzymatic activities, including beta-glucuronidases, azoreductases, nitroreductases, proteases, and many others, that can transform drug molecules in ways that profoundly alter their biological activity. The best characterized example is the microbiome's role in the toxicity of irinotecan, a chemotherapeutic agent used in colorectal cancer. The liver metabolizes irinotecan to the active form SN38 and then conjugates SN38 with glucuronic acid to produce the inactive, water-soluble SN38G, which is excreted in bile. In the colon, the beta-glucuronidases of the gut bacteria deconjugate SN38G back to the active, toxic SN38, which accumulates in the colonic epithelium and causes the severe, dose-limiting diarrhea that complicates irinotecan therapy. The bacterial beta-glucuronidase activity varies between individuals, partly explaining why some patients develop severe diarrhea on standard irinotecan doses while others tolerate them well. Inhibition of the bacterial beta-glucuronidase, a strategy being investigated in preclinical and early clinical studies, could potentially prevent this toxicity without compromising the drug's antitumor activity. Similar microbiome-mediated drug transformations have been described for digoxin, inactivated by Egerthella lenta in some individuals, sulfasalazine, activated by azoreductase-expressing bacteria, and many other drugs. As the clinical pharmacology field incorporates the microbiome as a drug-metabolizing organ alongside the liver and kidney, the prediction of individual drug responses will require characterization of the individual's microbiome as well as their pharmacogenomic profile. The phenomenon of taste adaptation, the rapid reduction in the perceived intensity of a continuous taste stimulus, mediated by the receptor mechanisms of the taste cells, is distinct from the long-term modulation of taste preferences that occurs over weeks and months of dietary change, which involves changes in receptor expression, central sensitivity, and the hedonic valence of specific food qualities. Taste adaptation operates on two timescales that serve different functions. The rapid adaptation. The well-known phenomenon of a strong flavor becoming less intense after a few seconds of continuous stimulation is a property of the taste receptor cells themselves, mediated by the calcium-dependent inactivation of the taste transduction channels and the depletion of the second messenger pools. It allows the taste system to remain sensitive across a wide range of stimulus concentrations by continuously recalibrating its baseline. The long-term modulation of taste preferences, the less rapid process by which habitual dietary patterns change what tastes good or bad, is a more complex phenomenon involving changes at multiple levels. The expression levels of taste receptors can change with dietary exposure. Chronic high sugar intake is associated with reduced sweet receptor sensitivity, requiring higher concentrations for the same perceived sweetness, a peripheral desensitization analogous to insulin resistance. In the central nervous system, the dopaminergic reward system's response to food-associated stimuli is modulated by habitual dietary patterns, with high-fat, high-sugar diets reducing the dopaminergic response to natural food rewards in ways that parallel the tolerance development of addictive substances. The hedonic valence of specific tastes, the degree to which sweetness or saltiness is experienced as pleasant, is also shaped by gut hormonal signals. GLP-1 receptor activation in the brain reduces the rewarding quality of sweet foods. The microbiome's production of short-chain fatty acids modulates the hedonic responses to different macronutrients through gut-brain signaling. Taste is not a fixed biological measurement but a continuously recalibrated, motivationally weighted assessment of the food's anticipated metabolic value. The concept of the gut as a barrier organ, one whose primary function includes maintaining the separation between the external environment of the gut lumen and the internal environment of the body, means that the digestive system's health is not simply a matter of nutrient absorption but of containing the potentially damaging contents of the gut within the gut. The word barrier in biology often implies a passive wall, a static, inert divider that simply stands between what should be separated. The intestinal barrier is nothing of the sort. It is a dynamic, actively maintained, multiply-redundant interface that continuously performs the selective discrimination of what the body should absorb from what it should exclude. The physical layers of the barrier, the inner sterile mucus layer, the tight junctions of the epithelium, the basement membrane, the lamina propria with its immune cells, each contribute their own exclusion mechanisms. The immunological layers, the secretory IgA, the intraepithelial lymphocytes that patrol the epithelium, the lamina propria macrophages that survey the submucosal space, provide active surveillance. The neural layers, the enteric neurons that regulate tight junction permeability and epithelial secretion, provide the dynamic adjustment of barrier function in response to physiological demands. And the microbial layers, the commensal bacteria whose competitive exclusion prevents pathogen colonization, the mucus-producing bacteria that maintain the mucus layer, the butyrate producers that fuel the epithelium, provide the living ecological dimension of barrier maintenance. The clinical importance of this barrier function becomes most apparent in critical illness. In the intensive care unit, the gut barrier is one of the first casualties of the physiological stress of sepsis, trauma, and major surgery, with bacterial translocation across the compromised barrier driving the systemic inflammation and multi-organ failure that are leading causes of death in the critically ill. The gut barrier is not the background context in which digestion occurs. It is a vital organ function in its own right. The composition of the diet during the first 1,000 days of life, from conception through the first 2 years, has disproportionate effects on the developing microbiome, the maturation of the immune system, the programming of metabolic set points, and the development of the enteric nervous system, making this window the most consequential for the long-term health of the digestive system and of the whole organism. The first 1,000 days, the period from conception through the second birthday, is a window of developmental plasticity during which the digestive system and its associated microbial and immune systems are being established, and during which the nutritional and microbial environment has effects on long-term health that are disproportionate to the duration of the window itself. The fetal digestive system begins absorbing amniotic fluid containing the food proteins, microbial products, and hormones of the maternal environment from approximately 17 weeks of gestation, providing the first antigen exposure that shapes the developing gut immune system. The mode of delivery determines the first microbial colonizers. Vaginally born infants receive the maternal vaginal and fecal microbiota, primarily Lactobacillus, Bifidobacterium, and the organisms of the birth canal, while cesarean-delivered infants are first colonized by the skin and environmental microbiota of the operating theater, producing a microbiome composition associated with higher rates of allergy, asthma, and metabolic disease. Breastfeeding provides human milk oligosaccharides, complex, indigestible carbohydrates that selectively promote the growth of Bifidobacterium species in the infant gut, along with maternal IgA, lactoferrin, and immune cells that support the infant's immune development. The introduction of solid foods at approximately 6 months begins the major transition of the microbiome toward the adult composition, with the diversity and composition of the complementary foods determining the trajectory of this transition. The microbiome composition at 2 years of age is substantially predictive of the microbiome composition in adulthood, and the immune education and metabolic programming that the microbiome delivers in the first 2 years shapes the individual's immune responsiveness and metabolic phenotype for life. The liver's production of C-reactive protein and other acute phase reactants during systemic inflammation, driven by interleukin-6 from the immune system acting on hepatocytes through the JAK-STAT signaling pathway, makes the liver an active participant in the systemic inflammatory response, and the chronically elevated C-reactive protein of low-grade chronic inflammation reflects the ongoing hepatic response to the gut-derived inflammatory signals of a dysbiotic microbiome. C-reactive protein, the plasma protein whose concentration rises rapidly and substantially during acute inflammation and infection, and whose measurement provides one of medicine's most widely used clinical tests, is produced exclusively by the hepatocytes of the liver, in response to interleukin-6 and other inflammatory cytokines released by the immune cells responding to infection or tissue injury. the liver's production of CRP and the other acute phase proteins, including fibrinogen, ferritin, serum amyloid A, haptoglobin, and complement components, is the hepatic arm of the acute phase response, the systemic reaction to infection and tissue injury that coordinates the body's defensive and repair resources. IL-6 from the site of inflammation reaches the hepatocytes via the circulation, activating the JAK1/JAK2 and STAT3 signaling pathway that drives the transcription of the acute phase protein genes. This acute phase response is dramatic in acute infection. CRP can rise 1,000-fold within 12 to 24 hours, but also operates at a lower level in chronic inflammatory states. The chronically elevated CRP of approximately 2 to 5 milligrams per liter, high-sensitivity CRP, the cardiovascular risk marker, reflects a persistent, low-level inflammatory stimulus reaching the hepatocytes. The gut is a major source of this chronic stimulus. The bacterial products that cross the compromised intestinal barrier in dysbiosis, LPS, bacterial DNA, flagellin, activate the hepatic Kupffer cells and portal endothelium to produce IL-6 and other cytokines that reach the hepatocytes through the intrasinusoidal circulation. The elevated CRP of obesity, of type 2 diabetes, and of unhealthy dietary patterns is, at least partly, the hepatic response to the gut-derived inflammatory signals of a compromised microbiome and intestinal barrier. The process of chewing, often automated and given almost no attention in the modern eating environment of distracted meals, has metabolic and digestive consequences beyond the obvious mechanical breakdown of food, including the stimulation of digestive secretion through the cephalic phase, the mixing of food with salivary enzymes that begin carbohydrate and fat digestion, and the slowing of eating rate that allows satiety signals time to register before overconsumption occurs. Chewing is the first act of digestion, and it is the one most subject to the environmental and behavioral influences of modern eating culture. The average modern person chews each mouthful approximately 12 to 15 times before swallowing, compared to the estimated 40 to 50 chews per mouthful in pre-industrial populations eating coarser, less processed foods. The reduced chewing of modern diets, with their soft, processed foods that require minimal mechanical breakdown, has physiological consequences at several levels. The cephalic phase stimulation of digestive secretion is proportional to the duration and intensity of oral processing. Abbreviated chewing produces less salivary amylase, less gastric acid, and less pancreatic enzyme secretion in the cephalic phase, reducing the preparatory state of the digestive system before food arrives. The incomplete physical breakdown of food by rapid swallowing reduces the surface area available for enzymatic digestion, potentially impairing the efficiency of the subsequent digestive phases. But the most physiologically consequential effect of rapid eating may be the temporal mismatch between food consumption and satiety signaling. The intestinal satiety hormones, GLP-1, peptide YY, and CCK, require nutrient contact with the intestinal mucosa to be released, and this contact begins only after the food reaches the small intestine, which is typically 15 to 20 minutes after eating begins. A person eating rapidly can consume substantially more food in this 15 to 20 minute window before the hormonal satiety signals reach the brain than a person eating slowly, explaining the well-established epidemiological association between eating speed and body weight, and the experimental findings showing that slower eating reduces total energy intake. The digestive system's production of serotonin by the enterochromaffin cells of the gut. 90 to 95% of the body's total serotonin makes the gut not merely a consumer of the neurotransmitter chemicals we associate with the brain, but a major producer. And the connection between gut serotonin, vagal afferent activation, and mood is one of the most biologically intriguing aspects of the gut-brain axis. The fact that most of the body's serotonin is in the gut, not in the brain, is one of those statistics that upends a popular narrative and opens a more interesting one. The serotonin in the gut is not a byproduct or a marginal player. The enterochromaffin cells of the intestinal mucosa produce and store vast quantities of serotonin in their secretory granules, releasing it in response to mechanical stimulation of the gut wall, to chemical stimulation by specific luminal contents, and to signals from the gut microbiome. The released serotonin acts on several target systems. It binds to the 5-HT3 and 5-HT4 receptors on the vagal afferent nerve terminals that course through the gut wall, generating signals that travel to the brainstem nucleus tractus solitarius, the primary recipient of vagal visceral afferents, and from there to the limbic system and cortex. It activates the 5-HT4 receptors on the myenteric plexus neurons that initiate peristalsis. It acts on the enterocyte apical membrane to modulate epithelial function. And the small fraction of gut serotonin that is not inactivated by the enterocyte uptake transporter, SERT, or by the enzyme monoamine oxidase enters the portal blood and circulates to the systemic tissues, contributing to platelet activation and potentially to peripheral serotonergic effects. The pharmacological success of the 5-HT3 receptor antagonists, ondansetron and related drugs, in treating chemotherapy-induced nausea and an irritable bowel syndrome reflects the central role of serotonergic signaling from the gut in controlling both gut motility and the perception of gut symptoms. The gut's serotonin is not simply the brain's serotonin misplaced. It is a distinct, locally regulated system whose outputs, both local and neural, are essential components of gut function and gut-brain communication. The digestive system is, at its most fundamental level, the biology of transformation, the system through which the matter of the outside world becomes the matter of the living body. And understanding it is understanding that every meal you have ever eaten has been an act of biological alchemy, and that the body performing this alchemy has been doing so, without acknowledgement, without credit, and without rest, for every day of your existence. Begin with the simplest version of this last fact: you ate today. In the body, this particular, irreplaceable body that has been with you through every version of yourself you have ever been, took what you gave it and began the process we have been exploring together for the past 100 facts. The amylase has been at work since the first bite. The gastric acid has been deploying since the cephalic phase anticipated the meal. The pancreatic enzymes have been disassembling the proteins and the fats and the carbohydrates into their constituent molecules. The villi have been reaching for the nutrients as they passed. The portal blood has been carrying the absorbed molecules to the liver, which has been sorting and processing and distributing and storing with the quiet efficiency it brings to every meal. The microbiome in the colon has been fermenting the fiber and producing the butyrate that feeds the colonocytes and the short-chain fatty acids that feed the whole metabolic system. The bile has been recycling through the enterohepatic circulation, used and reused again. And all of this has been happening in the 9 meters of living, specialized, continuously renewing tissue that runs from the lips to the last centimeter of the large intestine. 9 meters that have been working since before you could speak, that have digested 10,000 meals or more, that have extracted from the food of the world the precise molecular materials required to build and rebuild and maintain the body in which all of your experience occurs. The digestive system is not the background of your life. It is the foundation of it. Every thought you have had was powered by the glucose that this system absorbed. Every muscle that has ever moved you was built from the amino acids this system delivered. Every hormone that has shaped your moods and responses was synthesized from the precursors this system extracted from what you ate. You are, in the most literal sense, what you have digested. And tonight, as the body settles into its rest and the last meal of the day continues its quiet journey, that fact is not a burden but a kind of wonder. The alchemy continues. The river runs. The body keeps the faith. The digestive system you have been traveling through tonight is not separate from you. It is not a subject held at careful scholarly distance. The 9 meters of tissue we described are in your body right now, doing what they have always done, absorbing the last of today's meals, recycling the bile for tomorrow, letting the microbiome do its fermentation work in the colon while the rest of the body rests. The liver is processing. The mucociliary escalator is. No, that is the other system. The liver is processing. The migrating motor complex, if the stomach has been empty long enough, is beginning its housekeeping cycle. The gut-brain axis is sending its quiet signals in both directions, and somewhere in the rising melatonin and the falling cortisol and the lower metabolic rate of the approaching sleep, the gut is shifting from its postprandial mode toward the fasting mode, the mode in which the migrating motor complex clears the small intestine, in which the microbiome settles into its overnight fermentation pattern, in which the liver shifts from absorbing to producing. The digestive system, this second brain with its 500 million neurons, this largest endocrine organ with its dozens of hormones, this home to 70% of the body's immune cells, this river 9 meters long that transforms the matter of the world into the matter of the living body, is not a system you have been using today. It is a system you have been collaborating with, whether you knew it or not. You brought the food. It did the rest. the degree to which this collaboration has been successful, the degree to which the nutrients were absorbed, the pathogens were excluded, the microbiome was maintained, the barrier was preserved, is the degree to which you have been sustained for another day. This is relax with science, saying goodnight, and meaning it with the full weight of everything we have moved through together. The wonder of the digestive system does not leave when the words stop. It remains in the biology that is quietly continuing, the villi absorbing the last of the evening's nutrition, the microbiome fermenting the fiber, the liver doing its patient, unhurried work, the enteric nervous system running its autonomous programs, the gut producing its serotonin and its GLP-1 and its peptide YY in the quiet ratios of the resting state. You are not just someone who has heard about digestion tonight. You are the digestion. You are the 9 meters and the 500 million enteric neurons and the 38 trillion microbiome and the liver performing 500 functions simultaneously. And right now, tonight, all of it is resting, not stopped, never stopped, but slowing toward the quieter metabolism of sleep. The body has fed. The body is safe. The body is at rest. Let it be.